అడుగుతున్నాడు నేను 2 సంవత్సరాలు నువ్వు వస్తారా నేను చూసి ఆయన చూసి పట్టుకొని పొడిగి ఆయన చూసాను ఎందుకు బాగుంటాయండి 10 నిమిషాలు
Sir, good morning, sir.
Sir, Guntur. Okay,
sir. Good morning, sir. NRC Guntur responding, sir.
So, okay, sir. వినిపిస్తుంది.
Sir.
మీకు ఆడిబులే కదా?
మాకు ఆడిబుల్, సార్.
ఓకే సార్, ఓకే.
మీ
దగ్గర నుంచేనా ఇప్పుడు?
సార్, క్లారిటీ ఉందా?
క్లారిటీ ఉంది. ఈ రోజు మీ దగ్గర నుంచేనా?
అవును సార్, అవును.
ఓకే సార్. అయితే అనిల్ సార్ వస్తే అనిల్ సార్ ఇంటరాక్ట్ అవుతారు. లేదంటే మీరు మామూలుగా వాళ్ళని ఇంట్రడ్యూస్ చేసేసి కంటిన్యూ చేసేయండి.
సరే సార్, సరే సార్. సార్ ఇప్పుడు ఎలా ఉంది సార్? ఇప్పుడు ఒక్కసారి చెప్పండి.
ఇప్పుడు బాగుంది సార్, బాగుంది. ఆడియో బాగుంది.
బాగానే ఉంది కదా సార్?
బాగుంది సార్.
ఓకే సార్, థ్యాంక్ యూ సార్.
ఓకే సార్. సార్ ఎన్ఆర్సి తణుకు ప్లీజ్ రెస్పాండ్ సార్. సార్
క్లియర్ సార్?
క్లియర్ సార్, క్లియర్. బాగుంది.
ఓకే సార్, థ్యాంక్ యూ.
ఓకే సార్. హలో సార్, కాకినాడ ప్లీజ్ రెస్పాండ్. కాకినాడ ఒకసారి మాట్లాడండి. క్లారిటీ లేదు సార్, ఆడియో క్లారిటీ లేదు, ఒకసారి చెక్ చేసుకోండి. హలో ఆడియో కొంచెం బ్లర్ గా వస్తుంది సార్, బ్లర్ అవుతున్నట్టుంది, కొంచెం ఒకసారి చూడండి, ఆడియో క్లారిటీ ఉండట్లే. హలో సార్, ఓకే, ఆడిబుల్ ఆడిబుల్, కాకపోతే వాయిస్ కొంచెం క్లారిటీ అనేది మిస్ అవుతుంది. బట్ వినిపిస్తుంది వినిపిస్తుంది, అంత సమ్ క్లారిటీ మిస్ అవుతుంది, ఒక్కసారి చెక్ చేసుకోండి. ఓకే ఓకే సార్.
Hello.
Hello, good morning, madam.
Yeah, good morning. So I have joined, but you just let me know when everybody is here. I am keeping myself in mute option now and just waiting here.
Okay, madam.
Yeah. Okay then. Okay. How are you?
Fine, madam. How are you, madam?
Yeah, I am also doing great.
Okay, thank you, madam.
Okay then. Bye.
Hi, ma'am. Good morning.
Yeah, good morning. How are you?
Fine, ma'am. How about you, ma'am?
Yeah, I am also doing great.
Yeah. Thank you, ma'am.
Thank you.
Yes, madam. Sir came, madam.
Okay. Good
Yes.
morning.
Good morning, madam. Good morning.
Yeah.
How are you, madam?
I am fine. How are you?
Yeah, fine, fine, madam. Everything is going well.
Okay.
And one issue is pending, madam. Of course, from our side, that is KRPs. Actually, we requested all of you to support for their project and vet the project.
Okay.
Maybe you didn't receive these projects, I think. And once again, in front of you, I am requesting all our KRPs and other participants to please submit your proposals so that all the faculty, all the professors and scientists from University of Hyderabad will vet and they will recommend the projects so that we can get some funding. This is very important, going to be very important outcome of this training. That is, we are trying, madam, at our level best. I hope after the completion of this program, maybe we will receive some good projects also. So
Very
that
nice.
from your side, you can recommend and you can advise them if any changes are required.
Okay.
So that meanwhile, we are trying to give some sort of funding from our side.
Okay.
And later on, they can approach for DST, DPT, sir projects
Right.
like this.
Right.
This is
Some-
our motto.
Yeah, once they, generate some preliminary data with the funding received from your end, then that preliminary data will help to get bigger funding.
Yes, madam.
Yeah, because nowadays all the time they ask some preliminary data in order to have the potentiality that can be funded. So they can initiate the work with the funding from your side and get some kind of preliminary data, and with that they can apply for the higher funding agencies. Yeah, that will be good. We are here as we promised earlier. So there were small groups of 8 candidates versus 1 faculty, 1:8. That is how we made it. And in that group, they can approach and we will be there to guide. I mean, just to help. They
Yes,
know
yes.
everything, but we can just help them. No problem at all.
Yes, madam. Thank you very much.
Yeah.
I request all the KRPs and program coordinators, please submit your day-wise report at the end of the program tomorrow. And also, we will share the templates, that is certificates. Kindly arrange certificates at your level. I request the NRC principals, please sign the certificates and distribute maybe through online or offline. That is up to you. So tomorrow, immediately after this completion of this program, I request all the program coordinators and principals of NRCs, you can arrange validity at your end. So far, I think this is going well, and definitely there will be a transformation change among the faculties. And I request that please, all these are available in YouTube, all the online sessions. So kindly share this to your students also as well, because these are very high impact lectures. So the students will get benefit. Not only that, you please encourage them to write CO PG set so that most of our students can go for higher education in central universities like University of Hyderabad, Jawaharlal Nehru University, and Pondicherry University like this, because definitely these universities are very highly qualitative and institution of eminence. So in this background, this also should help our students. Our aim is to percolate all this knowledge to up to the level of students. So please also think about on this. Really, I appreciate the commerce KRPs. Commerce KRPs, they have already started online free coaching across India. They have grouped so many students from different colleges, and every day they are delivering, they are motivating, and they are giving coaching for civil services, different state services, and also PG entrance examinations. So this kind of approach, our KRPs also from life sciences can deliver this type of program. I think some of our faculty from zoology also doing this since long back. So other KRPs also can emulate this wonderful outcome. Maybe if all the participants joined, we can start, sir, because this resource person is already there. Yeah, please, sir. Sir, Srinivas Kara, Kakinada.
Good morning, sir.
Good morning, sir. Start cheyachu, sir, manam.
Yes, sir. Is over to NRC Guntur to introduce today's resource person, Professor Sunanda Bhattacharya from University of Hyderabad. Over to NRC Guntur.
Yes, sir. NRC Guntur respondents. A very good morning, everyone. I'm Sai Sudha, lecturer in biotechnology from NRC Guntur. On behalf of all the NRCs, I feel it a privilege to introduce our today's speaker, Dr. Sunanda Bhattacharya ma'am. Dr. Sunanda did her master's in biochemistry from University of Calcutta and PhD from Department of Biophysics, Bose Institute, Kolkata. She did her postdoctoral from Johns Hopkins University and Tulane University, USA. She joined the Department of Biotechnology and Biochemistry, University of Hyderabad in 2009 and still continuing. Dr. Sunanda's labs works on the role of HSP90 and its co-chaperones on genome stability. And also she is interested in understanding the mechanism of DNA replication in human malaria parasite, Plasmodium falciparum. Dr. Sunanda is a recipient of a prestigious Power Fellowship in the year 2023. She has guided over 7 PhDs and 5 are ongoing undergraduate credits. She has several national and international publications to her credit. With this very short introduction, I invite Dr. Sunanda Bhattacharya ma'am to deliver the lecture. Over to you, ma'am. Thank you so much.
Can you hear me?
Madam. Yes, madam.
Okay. So I'll be sharing, thank you so much for the introduction. So I'll be sharing my slides. Please confirm whether you can see it.
Yes, ma'am.
Yes, ma'am. Yes,
Okay.
ma'am.
Okay, so good morning, all of you. So today I'll be talking about, let me just, Today I'll be talking about assigning gene function by experimental analysis. I'll be focusing on gene inactivation. What are the different gene inactivation techniques are there? Okay. So whenever we want to understand the function of the gene, we need to have 4 different kind of experimental setup. The first thing is we need to inactivate that gene and try to look for certain phenotypic change. Suppose one particular gene has a role in DNA repair pathway. if we inactivate that gene and if we find that the cells are unable to grow in response to DNA damage, then we can tell that maybe that particular gene has a function in the repairing of those damaged DNA. So inactivation, how it can be done, that is the topic which I will be covering today. But only by inactivation, we cannot tell the gene function. It should have some other approaches in order to confirm the function of the gene, and which is either you overexpress that gene. Sometimes with overexpression, the function of the gene is very much prominent, and many gene novel functions has been identified by doing the overexpression of gene. Another important pillar is gene expression as a function of space and time. So if we want to understand the function of a gene, it is very important to know at what developmental stage it is getting expressed and what particular organelle within the cell it is present. So its localization and its time of expression can also lead to the understanding of the function of the gene. And last, another important aspect is this gene product is having what are the associated proteins with which it interacts. So if we can understand the protein-protein interaction, what are the different protein with which our gene of interest is interacting, then that will also give the idea that what will be the function of that gene. So being said that, as my topic of discussion today is gene inactivation, I will be providing 3 different aspects today. How to create non-functional gene. One is knockout of gene of interest from the chromosomal locus. Knockout means I am referring that that particular part of the gene is completely being deleted from the chromosomal locus. Then the second part is the gene remains as it is, but when gene is transcribing and forming the RNA, there are approaches how you can degrade the RNA and thereby you can inactivate that particular gene function. And the third approach, which I will be describing today, is the destabilization of the protein. So here the gene is present in the chromosome, the transcript is also stable, but as soon as the protein is being produced, we will be showing some approach by which you can destabilize the protein immediately and by which you can check the function of that particular protein. So first important thing I will describe what are the gene knockout strategy and which principle it is present within the cell, what kind of mechanism by which the gene knockout can be done. So in order to target a particular gene of interest within the chromosomal locus, you must take the advantage of the fact that cells know how to do the homologous recombination, because meiosis is the process within the cell where they utilize homologous recombination. So this is the inherent property of all living organism, because you know that whenever there is a parental chromosome and the maternal chromosome, they recombine within the homologous sequences and there occurs crossover, and that is how the recombined chromosome is produced. So that means there are some machinery within the cell which can recognize the homologous pair between the two DNA and they can form the homologous chromosome, homologous recombination between themselves. So, this is the schematic presentation, how the gene can be targeted. Suppose this is the chromosomal locus, you can see the blue color, and this is the target gene which you want to do knockout. So you are having a kind of cassette which is a plasmid which is having some marker gene, and you are having the upstream and downstream region of the target gene being cloned in that particular cassette. And if you transform this particular double-stranded DNA into that particular cell where you want to target this particular gene, the homologous recombination occurs between the upstream and downstream sequence of the target gene. As a result, the marker gene is incorporated within the target gene and the target gene is being knocked out. So this is the basic principle by which homologous recombination mediated knockout of the gene can be possible. So I will go little bit in detail. So in order to do gene knockout, there are certain things you need to have in order to start with. You should have a deletion construct plasmid. This is basically a plasmid. It is having all properties of plasmid and it has a yellow colored thing which I am showing as a resistance gene. So this is actually the marker which will allow you to tell whether the knockout of the target gene has occurred or not. Now in this plasmid, there are two multiple cloning sites and you have to clone the upstream and downstream sequence of the gene which you want to do the knockout. So suppose this is the genomic DNA and this blue colored gene is of your target gene which you want to do knockout. So you would basically need to clone the upstream part of the gene in your deletion construct and downstream part of the genomic DNA of your gene of interest in the deletion construct. And then you have to cut this plasmid so that, or you can PCR amplify this particular region so that the amplicon is generated where it is basically a double-stranded DNA with upstream and downstream of the gene of interest. So if you transform this piece of double-stranded DNA into the organism, there will occur the homologous recombination and by which you can see that blue colored gene is replaced by the yellow color of gene. So these are the different steps. Cloning of upstream and downstream of gene of interest in the flanking region of the resistance gene present in the deletion construct. PCR amplification of the cassette ranging up resistance gene and down and transform it into the cell and select in presence of neomycin or G418 sulfate to select the cell that carries the cassette. Because once this cassette has been incorporated, depending upon the marker gene which is present, you have to select those colonies which are retaining that cassette and that way you can consider that that gene has been knocked out. So basically you need to screen then to find out the knockout clones. Now this kind of recombination, homologous recombination for different organism, they have a different length of the homologous DNA need to be present for efficient homologous recombination to occur. As for example, suppose if you want to do knockout in a human cell line or in a parasite cell line, where the recombination requires a long stretch of upstream and downstream sequence. So you need to clone maybe 1000 base pair upstream or 1000 base pair downstream sequence in the plasmid DNA. However, it can be possible that in case of some organism, they require a very short stretch of homologous recombination sequences like as small as 40 base pair in order to occur the homologous recombination. Now, I will try to tell you that once you transform this piece of DNA, what are the events are expected to occur within the cell. So, when this particular piece of DNA is transformed within the cell, as we all understand that there occurs homologous recombination mediated gene replacement. So, this particular cassette being homologous, the upstream and downstream, this is integrated into the correct locus. However, in all the organisms, many of the organisms, they have another kind of repair pathway where it is called NHEJ mediated knockout. So it is non-homologous end joining mediated knockout. So what happens is that all the time within the organism chromosomal DNA, there occurs many inherent damages. Okay. So if there is any such damage present within the chromosomal DNA, then the whole cassette can be integrated in a locus, which is not the locus where you are trying to target. So these kind of generation of this kind of product also possible when you are trying to do gene knockout. So you can understand that the whole cassette is incorporated in some other part of chromosomal locus, and you need to therefore screen and try to take only those cells where the knockout has happened in the correct locus. What do I mean by that? So when we are trying to screen those colonies, you can understand that we are trying to understand what is the marker gene being present. So marker gene is present here also, marker gene is present here also. So if we grow, suppose in presence of G418 sulfate or neomycin, if we consider that the marker gene is kanamycin resistance or neomycin resistance respectively, then both these two groups of different cells will be found. But you have to understand that in this case, the knockout has occurred, and in this case, there is no knockout has occurred because the whole cassette has been incorporated in some other locus. So how to distinguish between them and how to understand only the perfect particular clone where the knockout has happened in the chromosomal locus. So I am taking some example how the knockout can be screened. And here I am talking about budding yeast, which is the, unicellular eukaryotic organism. And if you want to do gene knockout, I am giving a stepwise manner and giving an explanation how it is basically happening. So in yeast, generally, these marker genes can be used as different kind of auxotrophic markers can be taken like TRIP1 or URA3 because yeast is coming with different kind of mutant version. So you can see that this is a budding yeast, which is a TRIP1 auxotroph. What does that mean? It means that in its growth medium, we need to put tryptophan supplement for it to grow, because it cannot produce tryptophan by its own. So if you want to target a particular gene, say ASBA1, and you want to make a knockout of this gene, so this is basically the deletion cassette. And as I was mentioning that for yeast, the homologous recombination can occur if there is a homologous sequence of as small as 40 base pair long. So what you are basically doing, you are taking the deletion cassette and trying to PCR amplify the marker gene, which is tryptophan, TRIP1 you have taken. And in the PCR primer, you have added artificially 40 base pair upstream and 40 base pair downstream part of ASBA1. So that when the PCR amplification will occur, it will create the cassette flanked by the upstream and downstream sequence of ASBA1. So let's take some numbers. It will be easy to understand. Suppose TRIP1 gene ORF, okay, that is 1080 base pair long. And you have added, this is 1000 base pair long. And when you have done the PCR amplification with 40 base pair up and down, you have created a cassette which is of the size of 1080 base pair. Now, the next step, as I said, that after PCR amplification, you are doing the transformation. So when the transformation is being done, then due to homologous recombination, this TRIP cassette will be integrated into the chromosomal locus. So that we will be expecting that ASBA1 null is being created. And how we are selecting the cells? We are now growing these cells in absence of tryptophan. The growth media lacks tryptophan. So to start with, they were TRIP1 auxotroph. That means they are unable to produce tryptophan. So you are adding tryptophan supplement in the growth media. But once the gene knockout cassette has been transformed, you are selecting those particular clones by growing them in a media lacking tryptophan. So your purpose is, if this TRIP cassette has been incorporated, then TRIP1 gene product will be able to produce the functional tryptophan. So cells will be able to survive even in absence of tryptophan. So that is how you are selecting those colonies, which are the knockout colonies. Now, if you try to see exactly if you grow the cell in absence or in lack of tryptophan to select the cells, right away you cannot tell whether the knockout has occurred or not. As this picture is showing, suppose this is the chromosomal locus of SBA1 and this is the cassette. The cassette can go directly to some other part of the chromosome without going to the homologous site by NHAJ. So these group of cells will also be growing in absence of tryptophan. But in this group of cell, you can see SBA1 gene is retaining in its chromosome. So how to distinguish between these two populations? So you can try to understand that if you can get those colonies in order to screen them, isolate the genomic DNA, and if you use one of the primer which is upstream to the locus site and one of the conformation primer, so you can try to see that suppose in this case, you have taken a primer which is 200 base pair up of SBA1 and tryptophan reverse primer. So this PCR amplification will only possible if the cassette is going to the correct locus. As for example, if the organism is having, if that particular clone is having trip cassette incorporated in other part of the chromosome, then this forward primer will not be able to amplify this particular region of product because this other part of the chromosomal locus will not have this forward primer complementary sequences. So, in order to screen the colonies and try to find out the correct clone, all you need to do, you need to isolate the genomic DNA, use the forward primer, which is the locus specific, but little upstream region, and you use the reverse primer of the cassette. If you get some amplicon, then you can conclude that these are the clone where SBA1 gene has been replaced in its chromosomal locus with the cassette, right? But there is another possibility. Suppose this gene has been knocked out by this cassette, but still the cassette, the whole cassette, in addition to the particular locus, it has also been incorporated in some other part of the genome by NHAJ. So how to rule out that possibility? Because you do not want to create a condition where your gene is deleted, but again, some other part of chromosomal locus, that gene is also deleted, and you do not want to use that particular clonal population for your experiment. So this kind of selection of the colonies can be done by Southern blot analysis. So before starting that Southern blot analysis, how it can be done, I am showing here a schematic presentation how to interpret Southern data. So you all know that Southern blot analysis means you are isolating the genomic DNA and you want to understand the localization of a gene with respect to certain restriction endonucleases. So you can design which restriction endonuclease you will take. So suppose you are taking one restriction endonuclease which have this kind of sites where it makes the digestion. So this particular restriction enzyme, if you add in this double-stranded DNA, you understand that it will create a product of 5 kilobase, 3.1 kilobase, and 1 kilobase. Now, in order to see the Southern blot, you need to choose some kind of probe. And that probe is nothing but the complementary DNA, and the position of this probe is shown in this cartoon. So if you take this double-stranded DNA, digest it with the enzyme and use the probe A, and then you try to analyze the Southern blot data, your expected outcome will be, you will be able to see only 5 kb in the Southern blot because this probe is a radioactive probe. So basically, you are taking the whole genomic DNA, digesting with different enzymes, but then each of these digested fragment, you are allowing them to interact with this probe. So the probe is basically complementary to some part of the DNA. Where it is complementary, it will bind there, and being radioactive in nature, when you analyze that, you will see a particular band. So even if in this cartoon it is shown that there are 3 pieces of fragment being generated, if you use probe 1, it is very important which probe you are using, and you should know that where complementarity it is maintaining. So if the probe is A, then you expect the band to come 5 kb, because these 2 parts will not be complementary to A, and that will not be visible in your southern gel, southern blot. Similarly, if you use probe B, only 3.1 kilobase band will be found in the southern blot. If you use only C probe, only 1 kilobase band will be found. Now, suppose you are using the probe D, which is complementary to the midpoint of this thing. So then what will happen? Both this 5 kb fragment and 3.1 kb fragment, both will be showing the signal in the southern blot. Similarly, when you use probe E, you will find that both 3.1 kb and 1 kb fragment will be found in the southern blot. So being said that, let's go back to our problem. Our problem was, in order to do gene knockout, we have identified whether the gene has been deleted or not, but how to rule out the possibility that the cassette is not going and entering in some other part of the chromosomal locus. So in order to do that, we can do the conformation of the knockout by southern hybridization, which I am showing in this particular slide. So as you can see, here also I have shown one particular restriction enzyme sites. This is your favorite gene, which you want to do, want to create a knockout. Suppose this is your cassette. The cassette is having CanMax, that is kanamycin resistance gene, and it is having upstream and downstream sequence of your favorite gene. Now, when homologous recombination is happening, you can see that this cassette has been incorporated, and now a different restriction sites are generated because of this CanMax cassette, and these are the new restriction sites, recognition site of the same enzyme which is generated after doing the knockout. Now, if you want to confirm the knockout with respect to southern hybridization, and you are taking the genomic DNA of different clones and digesting it with this particular enzyme, and then using the probe which is made against CanMax DNA. Okay, so your probe is against CanMax DNA, and then you are running the southern blot. So suppose you have screened 4 different colonies, and in the southern blot, you are expecting different kind of bands. So you want to understand that which of these 4 different clones have the correct integration, and the integration did not occur in any other part of the chromosome. So let's see how to conclude. So you can see that if CanMax has been incorporated in the correct locus, the 2 fragments which will be generated, both of them will be complementary to the CanMax DNA, because the probe is in between these 2 fragments. So 4.5 kb and 3.1 kb is the expected band in the southern blot if the knockout has occurred to the correct locus and only in one point. But in case of 2, 3, and 4, you can understand that some other position you are finding the signals. So that way you can rule out that these number 2, number 3, and number 4 clones are not the desired one, and they are having the CanMax cassette which has been incorporated into some other part of the chromosome. So as it is not very interactive session, because I cannot take your question immediately, but I hope that I have been able to give you some understanding how to screen this kind of knockout clones and how to identify the one where the knockout has happened in the correct locus. Now I will briefly tell about the allelic exchange by homologous recombination. So suppose you have eliminated the gene of interest, and how to do that we have already described. Now you want to replace your gene of interest with a mutation in the gene. Okay, so how to create the mutant allele or replace the wild type gene with the mutant version, that I will be describing. So suppose you are working with a gene and you have understood that this is the function of the gene, and you want to make a point mutation in one of the active site region of that particular gene of interest, and want to create a cell where the gene will be replaced by one point mutation. So how to go about that? So first step is you make the site-directed mutagenesis in the plasmid where the gene has been cloned. So you can see this is a plasmid DNA where the gene is already being cloned, which is the wild type copy of the gene without having any mutation. And you want first to create a site-directed mutant where only one particular amino acid will be changed to the mutant version. So I'm not going in detail that how the site-directed mutagenesis is done. Generally, you need to create a long primer, which is about 40 to 42 base pair long, where the mutation will be incorporated in the middle part, and it will be perfect complementarity with the other part of the sequence. So suppose you want to have a mutation here from tyrosine to phenylalanine. So tyrosine, suppose it is present in the active site of the protein and you want to change it to phenylalanine. So you know the codon which will be coding for phenylalanine. You are giving the sequence of this phenylalanine coding sequence in the middle, and you are keeping 20 bases up and down, which are exact match with the particular sequence of the gene. So that once you do the PCR amplification, similarly the reverse primer will be there, the mutated double-stranded DNA will be created, as you can see here, that when the PCR will continue, it will continue from 5' to 3' direction, and there will be one nick present because the PCR product cannot be joined. Similarly, the forward primer, when it continues the PCR, it will continue the whole sequence keeping a nick here. But when it is doing the PCR amplification, this particular mutant primer is actually incorporating the mutant DNA, which is the PCR product which is being generated. Now, the whole concept lies into the fact that you are using the plasmid, you are doing the PCR amplification, taking the plasmid DNA as a template from a cell which is capable to do methylation of its DNA. So you can understand that any bacteria, you know that they use the DNA methylation. So after the PCR is done, you are basically trying to remove the parental plasmid from the pool of the PCR amplified plasmid. So the parental plasmid is all methylated. So if you digest this pool with DPN1, DPN1 targets the methylated DNA, and that is how the parental DNA will be cleaved, and you are left only with the site-directed double-stranded PCR fragment, and which you can transform into the E. coli. It can repair that small nick, and if you isolate the plasmid from here, you will be able to get the mutant DNA being created. So you wanted to generate a mutant allele, and just by doing site-directed mutagenesis, you can create this kind of mutation. Now it comes to the second part, how the wild copy of the gene in the chromosomal locus can be replaced with the mutant version. So previously we talked about how the wild type copy of gene can be completely removed by a knockout cassette. Now we will be describing how you can replace the wild type gene with the mutant version. It can be done with a two-step gene replacement, again the concept of homologous recombination. So suppose you have created your favorite gene, you have created one mutation. I have described this as yellow color, mutation is created. And this is the plasmid where that site-directed mutagenesis has occurred. Now you are adding this plasmid in the wild type cell. So wild type cell is already having the wild type version of YFG, and you have transformed this version. So there will occur the homologous recombination, and you can see that due to homologous recombination, this YFG will be having this whole cassette being integrated, and you will have this kind of fragment generated within the chromosomal locus of YFG. This is due to the homologous recombination, because you can see that this part and this part are homologous. So there will be a recombination by which this whole cassette will be incorporated. Now, so here you can see that in this case, in the chromosomal locus, you have both the wild type version as well as the mutant version. And there is a marker gene URA3, which is in between. Now, if you grow now the cell in presence of FOA, 5-fluoroorotic acid, then what will happen? Another round of recombination will be favored, because URA3 product is very toxic in presence of FOA. So there will be recombination between these two parts, because they are also very much similar in their sequence. Only one point there is a mutation. And that way, one population of cell will be created. As for example, I have given this cartoon to explain that this will loop out to make the homologous recombination, and one point will be created where this kind of product will be formed, and in another group of cell where this product will be retained, and the intermediate URA3 will be deleted. So in presence of FOA, if you now grow the cell for several generations and select the cell in a condition where there is URA3 present in it, then you will be able to select those mutant allele and the wild type allele. What you need to do now to do the sequencing and try to find out where is the mutation being generated. So basically, this is the way how you can replace the wild type gene with the mutant version. So being said that, I will also try to tell that there is a scheme for knockout when you are using the LOXP-CRE system. So why these kind of systems are required? First, let me tell you. So I have described that when the gene is required to be knocked out, you require some kind of cassette where a resistance marker gene is there, and you are selecting the cells in response of the property of that resistance marker gene. Now, suppose in a particular cell, you want to do serial gene knockout. You want to get the function of gene 1 and gene 2, gene 3, gene 4, gene 5. So you want to create a cell where it is having all 5 of the genes being deleted. So then your challenge is you have to find out 5 different resistance marker, because otherwise you cannot have that selection. Now, this is very much challenging. It is difficult to have lot of marker genes available as a deletion construct. So that is how there are options where one marker gene can be used over and over again. And how that can be done, that scheme I am discussing now. So basically, in this particular method, we will be describing that we are using the gene knockout system, and then we are removing the marker gene from the chromosomal locus. So all we have learned so far that the marker gene will be integrated into the chromosomal locus. That means this is already present in the chromosomal locus. For the next gene knockout, you cannot use the same marker gene. But suppose you want that, okay, I will use the same marker gene over and over again because in a same cell, I want to create 5 different gene knockout. I don't have more marker genes available. So how I can create a system where that marker gene can be reused again? So that can be done by LOXP-CRE system. It is a site-specific recombination. You can see what are the different applications of homologous recombination can be possible. So I'm giving an example. Suppose this is the chromosomal locus of HSC82. So this is the chromosomal locus and this is the ORF which you want to do knockout. So you have created a cassette where you have taken the upstream and downstream part of HSC82 and your marker gene is URA3. Now, in this cassette, there is an option that you have also a short sequence of LOXP being integrated. Now, what is this LOXP? Where from it is coming? I will describe it little late. Now, what is happening? Let's see. So, just by homologous recombination, which I have already described, this whole thing will recombine and this whole cassette will be incorporated. And you can see that you can select the colonies by growing in absence of URA3, because if the strains are to start with URA3 auxotroph, they will be now able to grow in absence of URA3 because this cassette has been incorporated. Now, you have tried to disrupt HSC82, that has occurred and the new cassette has been incorporated. Now, you want to create a situation where this URA3 marker is now eliminated from the chromosomal locus, so that for the next round of gene knockout, you can again use this cassette and you can use different upstream and downstream sequence. So how you are doing? You are taking advantage of these LOXP sequences which are present in the two sides flanking URA3. So this is a site-specific recombination system. If you overexpress CRE recombinase in that system, this will identify these LOXP sequences and they will recombine within themselves. So that you can understand pictorially, I can tell if this sequence you can consider as having 2 halves. This is also having 2 halves within the LOXP due to recombination with the help of CRE recombinase, because this protein can recognize only this LOXP sequence. Then this first half will recombine with the second half of LOXP and thereby the internal URA3 is eliminated. So you can see that in this way, HSC82 ORF is deleted and the internal URA3 is also deleted. So in this particular cell, if you want to delete another gene, you can utilize the same marker, just you change the upstream and downstream of your second target gene. So this is very, very useful for knockout of for serial gene knockout, one after another. So what is CRE-LOXP system? Actually, these sequences are taken from bacteriophage P1, which is having 13 base pair recognition site, separated by a 8 base pair spacer, and this is the CRE recombinase which can identify this LOXP sequence and they can do that. So you will be able to find that these kind of CRE LOXP sequences are being used to create the serial gene knockout, and another similar kind of systems are also possible which are known as FLP FRT recombination system. And these also recognize a particular sequence of 34 base pair long FRT, and the recombinase is here flipase. So with that, I will go to the from yeast to human cell line. If you want to create a gene knockout in mammalian cell line, the system is little different, and here we require 2 kinds of selection, not only the positive selection. Positive selection means you are putting a marker gene and as I have explained, that after the knockout is done, you are trying to understand whether this knockout gene has been incorporated in it or not. So you are growing the cell in presence of certain chemical, and if cells survive, that means positively selected, then you know that the gene integration has occurred. But in case of mammalian cell, we need to do both positive and negative selection in order to rule out the random integration versus the targeted integration. That means if the integration is happening via homologous recombination versus if it happens via non-homologous integration, that can be eliminated by using both positive and negative selection. And we will see quickly that how that can be done. So as for example, here you can see this is the chromosomal locus of your favorite gene, and it has an upstream and downstream sequence. This is basically a part of the deletion cassette which you are utilizing in order to do gene knockout in mammalian cell. Basically, it is having two components. One is it is having a neomycin resistance gene, and you have cloned the upstream part of your favorite gene and downstream part of your favorite gene, flanking the neomycin resistance gene. Okay, so there was in this plasmid, there was two multiple cloning sites. You have already cloned the up and the down part of the targeted gene locus. Now it has a second component which we will discuss now. It is a gene which is taken from herpes simplex virus, and the name of the gene is thymidine kinase. Now you see what is happening. Suppose if you now transform this double-stranded piece of DNA into the mammalian cell, it will have two outcomes. First, it can go to targeted integration. If targeted integration happens via homologous recombination, then you can see this up and down will recombine and the middle part will be incorporated into the chromosomal locus. So there will be no selection of this particular part of the cassette because this is other than the homologous part. It is just extending outward, right? But if it is going via random integration, then the whole cassette can be integrated in some part of the chromosomal locus. So there you can see the difference between these two outcomes is in both of them neomycin resistance gene has been incorporated, but in this random integration, additionally, you are having thymidine kinase gene of herpes simplex virus has been incorporated. So easily you can distinguish between these two types of cells where only targeted integration has occurred or where both the things has occurred. So you can see that pictorially, it is presented how the positive and negative selections are happening. So you can see that you have treated it with neomycin. That is the first selection, which is the positive selection. So I have given three different color combination because they are all in liquid, like the mammalian cell line. It is a liquid suspension. So three kinds of population is there after transformation. Blue is non-recombinant cell. That means you have done the transformation, but they do not have the cassette incorporated because transformation did not occur within them. Then you have this brown color where the recombinant with random integration. That means random integration means it has this cassette also incorporated in it. So that is the deep color. And you have another group where this is the violet color where the targeted integration has occurred. So once you transform, you have three kinds of population, but your desired population is only this color, right? So first you are doing positive selection. Positive selection means you are growing in presence of neomycin. So if the recombination, if they are non-recombinant cell, that means the blue colored cell where the cassette did not get integrated, then those cells cannot survive if you allow to grow them in presence of neomycin. So you are basically eliminating the blue colored cells. Now you have these two population. Now, in these two population, now the second thing what you are adding ganciclovir. Okay. So what is the function of this particular chemical? So this is a DNTP analog. So what happens is that if thymidine kinase of herpes simplex virus is getting expressed in this kind of cells due to random integration, then their substrate is ganciclovir. They will make it ganciclovir monophosphate, then diphosphate, then it will become triphosphate, and they will compete with normal DNTP, and they will be incorporated during DNA replication. While the DNA is getting replicated in place of the bases where DNTP is incorporated, it will have the similar kind of structure and it will be incorporated there, and it is an extreme poisonous condition and cell will immediately die. So you can see that if you use ganciclovir and thymidine kinase selection system, then the cell which will produce thymidine kinase, they will produce this kind of ganciclovir triphosphate, which will compete with normal DNTP, and thereby they will stop the DNA replication and the cell cannot survive. So basically, after treatment of ganciclovir, only those populations survives where there is this kind of random integration did not occur. So by using this kind of positive and negative selection, in case of human cell line, we can easily select between them. So being said that, I will tell another important method by which also utilizing homologous recombination, the gene can be removed or gene can be inactivated. So I will not have much time to talk about what is CRISPR. The whole meaning of CRISPR, if I see what is that, it is basically clustered, regularly interspaced, short palindromic repeats. So basically it has two components. One is clustered, regularly interspaced, and another one is short palindromic repeats. So like lot of other molecular biology research findings, this was first time identified in bacteria E. coli. So it was found that in E. coli genome, there are certain short repetitive sequence, short palindromic repeat. That is very important. Palindromic repeat sequences are present which are identical. So you can see that for E. coli, this is the kind of sequence which is present in short palindromic repeats. So idea is if they are transcribed, then the RNA will make a short hairpin loop because they are palindromic, so they will form the complementarity within themselves. But what was identified that there are some spacer sequences clustered, regularly interspaced. These spacer sequences were present also in E. coli genome, and people have no idea because what are these sequences? Because these sequences are pretty unique. They have no match, and every time it is new sequences. So they are unable to understand that what is the function of these sequences and where from they have come from, because they have no idea that what is the origin of these sequences. So it was identified during the time 2000 and late 1998 and 99 that these particular stage of sequences actually coming from the bacteriophage which infects the bacteria. So they found that these sequences are from bacteriophage, and basically it was identified that this is a immune system of E. coli. That is how these CRISPR sequences are being present. So let me tell you that what happens under normal condition if a bacteriophage attacks the bacterial cell. So it injects its DNA and try to replicate, make more phage particle and kill the bacteria and go out of this bacterial cell with more in number, right? But now with this CRISPR system being identified, it was understood that this particular phage when it enters within the DNA, then some other things are also happening. I am only showing these CRISPR sequences, which just now I explained that these are the short palindromic repeats, which are identical, and they are having some new sequences, unique sequences, which are called the spacer sequences. So these are actually the viral sequence which are infected, which has infected the bacterium in past. So what is happening is that when this part is getting transcribed, you can see that this green color portion is the viral part of the RNA, which is getting transcribed. And this is the palindromic repeat, which is forming this kind of double-stranded short hairpin loop. Now, there is another component which they have identified, that there is a CRISPR associated sequence. So CRISPR associated sequence, basically it encodes a protein which is known as Cas protein, and these proteins are having two different functions. As I said that Cas9 protein, suppose one such kind of Cas9 protein, it has two functions. One is helicase, one is endonuclease. So helicase function means this protein, once it binds to the double-stranded DNA, it can unwind, unzip the DNA. And it has an endonuclease, that means it can digest the DNA. So what is happening, this Cas protein is having certain structure where it can accommodate that CRISPR RNA which is being formed. So you can see that this CRISPR RNA can be bound to this Cas protein, and then they can search the new virus when it is infecting the cell, whether this green color portion is complementary to the virus which has infected to the bacterium. If it is found that this green colored particular sequence of RNA is having exact complementary sequence to that of the viral DNA, then this Cas9 protein can cause the degradation or the double strand break in the viral DNA, and that is how the viral DNA can be chewed up and virus cannot infect. So this is a kind of immune system of the bacteria by which they can eliminate the viral infection. So basically, when the virus infects, if it has infected previously, its own sequence is already being present within the bacterial genome. So if it is a different virus, already infected blue-colored virus, then that particular genome sequence is already being incorporated in the spacer. So as soon as that virus enters within the cell, this CRISPR RNA is being formed, and they are bound by this Cas9 protein, and they are going to search by unwinding the viral DNA and trying to see if there is a match. If they have a complementary sequence, then the Cas9 can chew the DNA, and that is how the DNA is getting degraded. Now the question will come that what if they do not have anything in their history? So then also the different kind of Cas associated sequences are there which can take the new part of the spacer derived from the virus, and it can integrate into the CRISPR sequence so that the memory has newly being created. So this is the whole CRISPR system, and you know that this system can be utilized in order to do effective gene knockdown. So it was proposed by, I will come back to this slide again. It was proposed by, Jennifer Doudna and Emmanuel Charpentier, who has got the Nobel Prize for CRISPR-Cas9 sequence identification, for this whole system. And what they thought that if they can make a chimera where the tracer RNA, is linked with the spacer DNA, this spacer DNA is nothing but the DNA sequence which you want to eliminate within the particular locus, then you can create this kind of chimera sequence, that is CRISPR RNA and tracer RNA chimera, And these chimera can be bound with Cas9 and it can be targeted where there is a DNA sequence where it is complementary, and then the Cas9 can cause the digestion of the DNA, thereby the break in the DNA is being created, and it can be repaired by non-homologous enjoining so that some kind of insertion or deletion being created, and that causes the inactivation of the gene. So basically the concept is like that, that you can have a plasmid where this kind of tracer RNA, which is also known as guide RNA, is being produced, which has a typical association with a Cas9 protein, but it is in frame with a small piece of RNA, which is known as the RNA sequence, which you want to target at a particular part of the chromosomal locus. Suppose this is the double-stranded DNA and this is your favorite gene, and you want to have a knockdown situation. You want to make inactivation of this gene. So all you need to do, you need to clone a very short piece, 20 to 21 base of DNA. You can clone it in a vector, and this DNA, when it will produce the RNA, it will be linked with this guide RNA. And in this system, if Cas9 protein is also being expressed, then Cas9 will be bound to this guide RNA, and just like it does in case of the viral DNA, it can try to find the match where this particular sequence is having complementarity, and if there is a match found, then Cas9 can cause the digestion in the target DNA, making a double strand break. So this is in a very simplified fashion to explain that how CRISPR-Cas9 system can be utilized in order to do the gene double strand break in a particular locus. And once the break is created, automatically cell will try to repair this damage. If they involve the NHEJ mode of repair, then either they can insert new bases so that the total frame of the DNA is changed, so it is now having lot of mutation, or it can delete some part of the region because that is how NHEJ causes indel mutation, insertion or deletion, and thereby you are creating a mutation. So nowadays, the CRISPR system also can be utilized in order to do gene therapy, where there is a third component. So the first component is making the spacer, CRISPR RNA, which is the sequence where you want to target the gene of interest, and it is linked with the tracer RNA. You have another component which is expressing Cas9 within the system. And the third component is if you have incorporated a homologous donor DNA, that means this is the donor DNA which you want to incorporate, but it has a flanking upstream and downstream sequence where you want to create a cut by CRISPR-Cas9 system. So if you have this three component system, then this donor DNA will be integrated, and that is how you can put you can put a new gene in a targeted manner, and that is how it is known as a very fantastic gene editing system. You can incorporate one gene, you can delete one gene, and this is a very useful technique in order to do that. So this kind of plasmids are also available depending upon which organism you are working, and Cas9 expressing protein expressing plasmids are also available. So all you need to do, you need to just clone this short piece of fragment, and then once it is producing the RNA, you need to transform that into that particular cell line where you want to create the knockdown. You want to also need to express Cas9 in that organism, and then rest of the thing they will do by themselves. Okay. So now I will talk about the different other methods which are there in order to do the gene non-functionality of the gene. How to create non-functional gene. One part I have described so far. I have discussed that by homologous recombination method, how we can eliminate the gene from the chromosomal locus. Now we are not involved in the gene elimination. Gene is there, in the chromosomal locus, but once it is transcribing and forming the RNA, what are the techniques available to degrade the RNA immediately after it is getting transcribed? So there are 3 different steps how this can be achieved. One is, first you need to design a long double-stranded RNA which is having complementarity to the mRNA which you want to degrade. So you have a favorite gene. That favorite gene has transcribed to form your favorite RNA. You want to eliminate that RNA from the system. So you are making a double-stranded long RNA, and these double-stranded long RNA you have transformed or you have somehow incorporated within the organism. Okay, and these double-stranded RNA are known as also silencer RNA, and they are basically 21 to 23 nucleotide short fragments which can do this kind of degradation of the mRNA. But we will now try to understand how this can be achieved. So in the first step, you are giving a long double-stranded RNA, and if you add a particular enzyme known as dicer, they will dice it, and you can see that they will form this kind of siRNA, short interfering RNA of 21 to 23 nucleotide long, which is known as small interfering RNA. So dicer is basically having 2 different domains by which it can digest. It contains 2 RNAs, 3 domains by which it can digest the long double-stranded RNA to form this short, small interfering RNA. If we see what is the structure in siRNA, so they have a very definite structure. They are 19 nucleotide duplex and having 2 to 3 nucleotide overhang at the 3' end. Okay, so this should be the structure of, it is a very defined structure. This should be the structure of siRNA. So once this siRNA is created, it is taken by some complex which is known as RNAi silencing complex. So now you have to understand that all living organisms do not have this silencing machinery in it. So if you want to do knockdown by this approach, first important thing you need to know whether that particular organism is having RNAi silencing complex present in it. Remember in the first step when I discussed about homologous recombination mediated gene knockout, I told that homologous recombination machinery is present in all organisms because every organism undergoes meiosis. And that is one of the major thing has to be present. But this RNAi silencing complex is not present in many organisms like Saccharomyces cerevisiae, like Plasmodium falciparum. So you can understand in those systems, you cannot do the gene knockdown using siRNA approach. Because this complex is actually what it is doing, it is unwinding this siRNA. So if you can see that this is the particular mRNA which is formed and you want to target this mRNA. So you want to cleave this mRNA so that it cannot go for further protein. It cannot go further for translation and protein translation. So this is the siRNA duplex, which is basically the double-stranded RNA. And you can see that this part is perfectly complementary to one of the part of this mRNA. So what is happening? This silencing complex unwind this siRNA, takes the complementary pair and then search for the mRNA where they find the complementarity. That antisense strand then can bind to that, and immediately this complex can cause degradation of the mRNA. So using this kind of siRNA technology, you can eliminate the transcript once it is being formed. So now the question is how to make double-stranded RNA. There are many different ways. So suppose you want to make double-stranded RNA, you have the gene of interest, you can do the PCR amplification, and during amplification, you are adding T7 promoter sequence artificially in one of the forward and reverse primer, so that after PCR amplification, you have generated T7 promoter region incorporated in upstream and downstream sequence of this PCR amplicon. So once this PCR product is formed, you are taking it in a test tube and you are performing in vitro transcription. That means you are adding T7 RNA polymerase. So you can see that it has the T7 promoter sequence, which you have artificially incorporated, and that promoter will be recognized by T7 RNA polymerase, and it will cause the mRNA in both the direction, because you have you have incorporated T7 promoter both in this way as well as in this way. So you can see that it will form the double-stranded RNA, and this is how you can make the double-stranded RNA. And there are many ways how you can do the delivery of this double-stranded RNA within the cell. You can use the liposome-mediated delivery, and that is how double-stranded RNA can be incorporated within the cell. Now, so there are many different research, and I mean, this is very much technical part. We do not need to worry about these things much, but I'm just giving an overview. So either you are producing a long double-stranded RNA in vitro condition, this is suppose the cell, and you can see that either you can do microinjection, and if you do that, dicer is present in the cytoplasm of those organisms where siRNA silencing complexes are present. So it cuts it and makes the siRNA. Or siRNA can be created outside, and then by transfixion, it can be incorporated. Or in other words, you can have a plasmid incorporated within the cell where sense and antisense DNA, antisense sequences are kept in presence of some spacer, so that when the transcription occurs, it makes this kind of short hairpin RNA, which is getting translocated from nucleus to cytoplasm. Then it acts by dicer to produce the siRNA. So there are many different ways siRNA is produced. And once siRNA is produced, it is bound by that silencing complex. It unwinds this, and the complementary or the antisense of RNA is being carried, and it can search for its template where it can be complementary, and wherever that mRNA template is there, it will bind that position, and then the silencing complex can degrade or chew that mRNA. So that is how you can see that the expression level, the expression is lost. Expression means that mRNA is not stabilized. So you can create this situation in order to inactivate the gene by removing the transcription, removing the transcripts, and you can understand the function of the gene in that context. Now, many times it can occur that you are trying to eliminate the transcript of a particular gene, but that is very essential for the cell survivability. So if you create a siRNA mediated knockdown approach, it can be possible that it is very toxic to the cell because some essential gene will be lost. Okay, so in order to avoid that, there are options by which you can have an expression system which is inducible. What is the meaning of that? The meaning of that is you want to create a siRNA, but that you want to create not constitutively. So I have described here that you can have a system, you can put one kind of vector, that is plasmid, which is having some Pol3 promoter, and in downstream of that you have the sequences by which you will create the shRNA. Now, if shRNA is created all the time constitutively, then it will be forming the siRNA and inactivate the gene. And if the gene is very essential, it will be very toxic to the cell. So cell cannot, host cannot survive because it is so essential. So you do not want these to be produced all the time. You want to create a situation where only when you want to do the experiment, then you want to induce the expression of this transcription. You want to induce the expression of this product. So how that can be done? You can see here, there is some kind of manipulation done in the upstream regulator sequence. So this is a promoter. You can see Pol3 promoter, recognizing promoter, and followed by there is a TOT operator system. This is a small cis regulatory system which is incorporated into the vector. Within that cell, under normal condition, you need to express TET repressor, tetracycline repressor. So tetracycline repressors have a huge affinity towards tetracycline operator. So they will bind to that operator, and that is how RNA Pol3 cannot create the transcription under normal condition. So you are targeting a particular sequence, but you are keeping it in a silencing condition under normal condition with the help of TET repressor, TET operator system. But when you want the formation of this kind of short hairpin RNA, then you are adding doxycycline within the cell. So doxycycline will bind to this TET repressor and change their conformation so that TET repressor cannot bind to the TET operator like before, and then the promoter is free for RNA polymerase 3 to bind, and then the target sequence can be transcribed. So this is extremely tight on-off regulation, and it is ideal for the cases where gene suppression may be lethal. So you can understand that this particular siRNA creation can be done in an inducible condition where you can create the situation only when you want to perform the experiment due to the toxicity. Okay. So now this is actually something I just want to tell, that when people are trying to add the double-stranded long RNA to do the silencing of the transcript in case of mammalian system, they were not successful for very long period, because in mammalian system, these double-stranded RNA, if being incorporated within the cell, then they causes some kind of response, and that response is actually RNA-mediated protein kinase. There is a particular protein, RNA-mediated protein kinase, that is getting autophosphorylated, and that causes phosphorylation of EIF2 alpha, and it causes blockage of protein synthesis, and on the other hand, it causes interferon production and causes death or apoptosis. So basically, why mammalian cell is having this kind of system? Because this is a defense system against the viral infection. So you want to do gene knockdown by double-stranded RNA, long double-stranded RNA, and people are failing because inside the mammalian system, whenever you put that double-stranded RNA, it is activating as if it is a viral infection, and they causes the cell death because PKR is activated, and then this downstream methods is going on. So it was Dr. Thomas Taschel who has found his groundbreaking experiment, where he has found that longer double-stranded RNA, which lead to the non-specific type 1 interferon response, can be avoided if RNAi can be done with a very short 21 to 22 nucleotide RNAs. So he has shown that in mammalian system, this silencing can be done where if you can only put short 21 to 22 nucleotide long siRNA, not that big long RNA, double-stranded RNA, then this kind of response can be bypassed, and you can target the specific mRNA by using this approach. Okay. So last 10 minutes I will just describe the last part, that is how you can inactivate the gene by destabilize the protein that is translated from your gene of interest. So in order to do that, there are certain options. Suppose this is the gene which codes the protein of your interest, POI. You can create a situation by which you can put a destabilization domain encoding sequence in frame to this POI at the chromosomal locus. So what is destabilization domain? Destabilization domain is FKBP12, which is basically is FK binding protein 506. And it was found that this particular domain, if a mutant part of this domain is incorporated in the N-terminal part or C-terminal part of the protein of your interest, then the protein is extremely short-lived and it is degraded. It is having very short half-life. So you can understand that every protein inside the cell, once it is created from ribosome, it is having certain half-life. After that, it is ubiquitinated and it is going via proteasomal degradation. So all proteins are having different kind of half-life and different amount of stability. It is observed that by some experiment, which was first time performed by this group of scientists, it was published in Cell 2006, where they have shown that this is the property of this protein. If there is a mutation in it being created, this particular protein has a very short half-life. So what they thought that if this small protein, its total size is only 12 kilodalton, if the gene encoding this protein can be fused with the gene of interest, which you want to eliminate from the cell, then when this part will be transcribed and translated, it will form this kind of fusion product where destabilization domain is linked with protein of interest, and that will be immediately getting degraded. And so you can create a situation where your protein of interest is not present. That is, you have inactivated the gene of interest. However, this destabilization domain has a binding site for a small chemical called ligand. If you add that ligand, then the conformation of this DD has changed and it is having no immediate degradation. And then the protein can be stabilized. So you can have a switch on, switch off system by adding this kind of ligand in it. And if I tell what is the structure of this ligand, which when binds to this DD domain does not allow the degradation, this is the total structure of this particular ligand. So you can see that they are having two different options. One is very popularly used, which is known as shield 1. So you can see this is the small chemical where this is the R group. So in case of shield 1, the R group structure is this. And in case of SLF, the R group structure is like this. Okay. So you can see that if you add shield 1 present within the system, then the protein is stabilized. If you do not add the shield 1, then the protein is immediately degraded. So let's see how that can be taken. So this is the picture taken from that Cell paper 2006, which just now I described. They have taken a mutation of this destabilization domain and they have fused it with YFP, that is yellow fluorescence protein, and they want to understand the stability of this protein in presence and absence of shield 1. So if you see that they have grown the cell in presence and absence of shield. So you can see that in this condition, there are 4 different conditions. All of them is having L106P, okay, leucine change to proline, this mutant form of FKBP, which is fused with YFP. And this is acting as a loading control. That means 4 different conditions were taken. In one condition, they have not, I'm talking about MG132 little later. So let's see these two len. So you can try to understand that shield 1 is present and here you can see shield 1 is absent. So when shield is present, you can see that YFP is giving the signal if you probe it with anti-YFP antibody, you can see by the Western blot that the protein is stabilized. When you do not give the shield one, then you are seeing that protein is completely degraded, right? But to show that the protein is there in this particular condition, they have probed it with another loading control, which is telling that in this lane protein is present, but specifically YFP is getting degraded. Now, why YFP is getting degraded? They have tried to use proteasome inhibitor. So proteasome inhibitor is Mg132, and that is telling that this is eliminated because of proteasomal degradation of the protein, because in a condition where you did not give shield, but you have added Mg132, then this protein can be observed. So how this experiment is done, it is written here. This is the particular cell line expressing this system, and they were treated with 1 micromolar shield for 24 hours. Cells are then washed out and treated with Mg132 in presence and absence of shield. Immunoblotting was performed with anti-YFP antibody. So basically the bottom line of this particular technique is whatever protein you want to target, you want to eliminate that, you can put the destabilization domain in frame in the chromosomal locus. So once that protein is formed, you try to keep the cell in presence of shield 1 added in the medium so that the protein will be present. But when you want to do the experiment, then you wash out shield from the media. So as soon as the shield 1 being washed out, you will find that the protein is not present within the cell at all. So you can create a situation where if shield 1 is present, the protein is found. If the shield is absent, the protein is eliminated. So this is the very strong technique by which you can also also make the inactivation of the gene by targeting the stabilization of the protein. So this is also another way they have shown, as it is a yellow fluorescence protein, when they are not adding shield, there is no fluorescence found because it has been destabilized, it is not present at all. When 1 micromolar shield is added, then the protein is stable because it is giving some kind of yellow fluorescence. So then they have compared that, they have taken this YFP fused with L106P destabilization FKBP, and they have found that after they have removed the shield, everything is written here, how much time it takes for the protein to destabilize. Because I am telling that when you are using that system, to start with, you grow in presence of shield, and then you are eliminating, removing the shield. So after you wash out the shield, you are doing the Western blot at every hour interval and trying to find out how much time it takes for the protein to completely degrade. So as they have done the Western blot for 48 hours, they are finding that at second hour only, there is about, more than 50% reduction in the total protein level, which in fourth hour, it is completely being eliminated. So they have compared this technique with that of the siRNA mediated gene inactivation. So they have used lamin A and lamin C, try to do the knockdown by siRNA, which just now I mentioned. And when they are trying to see the level of the protein, they found that if it is knocked down by siRNA, even after 24 hours also, there is a faint impression of protein remains. So they are trying to propose that DD-mediated degradation shows faster kinetics than RNAi-mediated silencing. But you understand that you cannot really compare with YFP with some other gene. because every gene can show different effect. But when you will be using siRNA mediated silencing, you need to do this kind of experiment to find out how many hours after addition of siRNA it takes for the protein to completely eliminate it from the system in order to design your experiment. Because if you just add siRNA within 8 hours, you start doing the experiment, the protein is still present as that of the 0 hour. That means it is taking some time to get the degradation of the mRNA and degradation of the protein, which is reflected here, because if mRNA is degraded, the protein will be eventually less. So this is how you need to do the experiment first to understand that how much time it takes, and then only you can continue with that. So with that, I have covered 3 different aspects by which gene can be inactivated, and this is my last slide. And I'll be happy to take few questions. I think it is just 11:30. So, are you there? Yes.
Request all the participants, various NRCs, Professor Nodal Resource Centers.
Actually,
Excuse me,
the
ma'am.
voice is breaking very much. I'm not able to hear you properly. Can you?
Madam,
NRC, please
Yes.
check your audio and interact with Professor Sunanda Bhattacharya. NRC, please check your audio once, madam.
excuse me, ma'am.
Yes.
Is there any DD means protein destabilization for oncogenes which are expressed in HeLa cells?
No. See, here I am not touching any property of oncogene getting destabilized. I am touching the part how you can utilize destabilization domain method in order to destabilize any protein of your choice. Suppose you want to destabilize one particular oncogene from the cancer cell to understand its function. So that time, what you need to do, you need to put this destabilization domain in frame with that oncogene, so that once it is produced, it will be degraded in absence of shield. So every protein has a different half-life. So I am not touching that which protein is short-lived, which protein is long-lived, and the property of that. It is a generalized approach. It can be added to any gene product if you put it either N-terminal or C-terminal end of that protein. In absence of the chemical, it will be degraded.
Thank you, ma'am.
Okay.
Srivakodam. Srivakodam.
Hello? Yes, I can hear you. Tell me.
Srivakodam.
I can't understand. Can any other person just mention it, what he is asking? Madam, can you repeat his question? Madam, your voice was very much audible. Who asked me first time? Her voice is very good. I mean, it is audible to me. Can you please repeat his question?
Sure, madam. We will repeat the question for you. NRC ask the questions. Is it audible, madam?
No, it is breaking very much. The first madam's voice was very clear. Can she repeat the question?
Sure, madam. We will plan like that only, madam. Is it okay, madam?
I am unable to open that. Just a minute. Yes, tell me.
Request all the NRCs All the other NRCs, please interact with the Professor Sunanda Bhattacharya madam. There are no queries. Then I request NRC Guntur to propose vote of thanks. For NRC Guntur.
Ma'am, on behalf of all NRCs, I thank you for your insightful lecture, ma'am, on assigning gene function through experimental analysis and gene inactivation. Your expertise and lucid explanation made a complex topic very easily understood, ma'am. We understood how homologous recombination using deletion, construct, transmit, and screening the gene knockout cells, and we also understood the site-directed mutagenesis process and how to replace the wild-type gene with the mutant version, ma'am. And we also clearly understood the serial gene knockout systems using recombinase. And you also has given clear explanation about positive and negative selections, ma'am. And you have briefly and clearly explained the CRISPR-Cas9, how to use it for gene editing, ma'am. And we also understood the synthesis of double-stranded RNA using T7 promoter and use of this for mRNA silencing, ma'am. And it's a very new topic for us, that is destabilizing domain, using destabilizing domain, how to stop the functioning of the protein, ma'am. So at the three levels, that is genome level and transcriptional level and translational level, how to edit the genes, it's very clear, ma'am. Thank you very much for your wonderful lecture, ma'am. And your explanations left no room for confusion. It was brilliantly explained, ma'am. So thank you very much, ma'am.
Thank you very much. I'm very happy to deliver this lecture to you all. And I wish that in future, if any kind of these kind of things are required, you please let me know. I'll be there.
Thank you,
Thank you,
ma'am.
madam.
Ma'am,
Okay,
thank you, ma'am.
okay. Can I leave now?
Yes,
Okay, okay. Thank you very much. Thank you, all of you. I'm seeing many of you after a long time.
yes,
Yeah.
ma'am.
Yeah.
Yes, ma'am.
Thank you, ma'am.
ప్రిపేర్ అయిపోయాను సార్, చెప్పేస్తాను. చెప్పేస్తారా? వద్దులేండి, మీరు చెప్పండి. చెప్పలేదంట మాకేం చెప్తాం, మళ్ళీ అవన్నీ చూస్తారు. అవన్నీ తీసి అందరూ కావాలి. NRC Visakhapatnam, are you ready, sir? NRC Visakhapatnam. Professor Arun Kumar, sir, good afternoon, sir. We will wait for another few minutes, sir. Am I audible, sir? Arun Kumar, sir. Professor Arun Kumar, sir. Am I audible, sir?
You're able to hear me?
Yes, sir. It is audible, sir. It is audible.
Yeah. Thank you.
We will wait for
All
another.
of you.
Yes, yes, sir. 5 minutes.
You have to tell me if you're able to see the screen. Are you able to see the screen?
Sir, sir, your screen is there, sir, but the presentation is not there, sir. Your screen is visible, sir.
Yeah.
Ah, share the presentation, sir. Your screen is there. All NRCs are visible there as tabs.
I'm not used to this Vesco thing, so let me see what has to be done here. Do you want to share?
At the bottom, there is something like share icon is there, sir. The address bar, something like that. Bottom. Just click on
So allow WebEx to see your screen. Under that category, I'm getting Cisco, WebEx, Microsoft PowerPoint, and entire screen.
not like that, sir. At the bottom of the screen, there are mute, start video, share is there, sir. Right hand. Next to right hand, there is a share icon is there, sir.
Now, can you see?
Your screen is sharing, sir, but not PowerPoint is not sharing. Screen is there.
Okay, let me try again. 1 minute.
Just at the bottom of this, your screen,
Yeah.
there are 4 tabs, sir. One is mute, next one is start video.
No, I get that. There is one called as share, right?
Yes, exactly, sir. That is enough, sir.
Yeah, that I'm already clicked. It says you are sharing your screen.
Then you maximize the presentation.
Pardon?
Maximize your presentation, sir. That is full screen.
Maximize.
You go for the full screen of your presentation, sir. Next.
Now?
Let's change the layout, sir. At the bottom of the screen, layout is there, sir.
No. Now can you see the screen? Now, because I am able to see. Are you able to see or no?
No, sir. At our end, only tabs of all NRCs are coming. That's the only coming from our end. Still, it is connecting. It is prompting like connecting. Click share to share content meeting.
Share.
I'll get back to you, sir. I'll ask a technical
Yeah.
person. See, please.
Okay.
Just a minute.
How about now?
Not sharing.
You're able.
Still not sharing.
Still
Okay, not sharing.
No, you should. Why don't you put it on Google thing? It will be more easy, na?
Actually, this platform is from the head office, sir. CC office, Vijayawada.
Okay, fine.
We all will come from various centers from Visakhapatnam to
So you are able to hear me, right? Let me tell you what I'm doing exactly. I have gone to this share. I've clicked the share.
Yes, sir.
It says it's connecting.
Yes, sir. Complete that connecting dialog box. Saki.
See, I'm getting a message here that I'm sharing the entire screen. I don't know why you're not able to see.
Sir, Kumar sir,
Yes.
is it possible to share the PowerPoint through WhatsApp or then I will display from here from my end?
I think it's the PowerPoint is too big to be shared, but anyway, I'll try once.
Sir, it is asking share your window or share maximize window or tab. Still it is connecting. It's connecting, sir.
Okay.
After the dialog box is closed, you may share, sir. It seems.
But it's wasting the participants' time, na? This is a problem with the net connectivity, you think?
Yes, sir. It seems the connectivity problem with this.
Sorry, let me do one thing. Let me send you the PPT. I'm just attaching there. Yeah, you try to
That is better,
sort of
sir. That is
open it
better.
from there. Yeah, open it from there.
Please, sir, revert to the mail which I have sent to you, sir. Yes, sir.
See, this Google Meet links are very quick to create one and share with ease. Actually, other than that, we are not much used to the other kind of uh, links.
Sir, have you sent the mail, sir?
Yeah, I'm sending. It's uploading very slowly. I think it'll take a minute more. Just a minute. Sir, I've sent it to you. Any desk ఉంది నా ఉన్నట్టుంది. పంపించేసాను నేను ఆల్రెడీ. ఒక్కసారి చూస్తాను, చూడండి ఒకసారి మీరు. నేను ఎనీ డెస్క్ చూస్తాను. You got the presentation?
Yes, sir. You got the presentation, sir.
Yeah, that's why it took so long time.
AP teachers, that means.
AP teachers అని పెట్టేసేయండి. మీరు ప్రొజెక్ట్ చేస్తారా? యా, ఓకే.
Is MDP 149,094. That one is, sir. Meanwhile, I request NRC Women's Visakhapatnam to introduce this afternoon session resource person, Professor Nodal Resource Centers, for the all the participants, the various NRCs are other friends. Both the NRC Visakhapatnam, please introduce this session resource person, Professor Nodal Resource Centers. NRC Visakhapatnam, please respond once. NRC Visakhapatnam, over to NRC Visakhapatnam. Hello. I request the faculty from NRC Visakhapatnam introduce Professor Nodal Kumar sir.
Hello.
Yes, sir, you are audible to NRC Visakhapatnam.
Okay, sir.
Hello. Sir, am I audible, sir?
Yes, madam.
Yes, sir.
Audible, madam. Proceed, madam. Rekha, madam.
Yes, sir. Yes, sir. Sir, I deem it a great privilege to introduce Dr. Kota Arun Kumar Garu to all the participants of TOT. Dr. Arun Kumar has been the alumnus of University of Hyderabad since his master's and finished his doctorate at UOH. He did his postdoc from New York University School of Medicine, USA, where he studied pre-erythrocytic stages of Plasmodium using rodent malarial models. Now he is working as assistant professor in animal biology, School of Life Sciences, University of Hyderabad. His work continues in Plasmodium itself, where he is trying to prevent clinical malaria, prevent prevent the transmission of malaria, which has implications in producing attenuated vaccines. Sir, I think you must be in head over heels in awe with Plasmodium, and we, the participants, are ready to hear your immense knowledge on Plasmodium, sir. Over to you, sir.
Professor Arun Kumar, sir. This the presentation is going to come out, and sir, is it visible to you, sir?
Srinivas sir, full screen mode pattern sir, slideshow mode.
Sir. Sir, Arun Kumar, sir. Hello, sir. Hello.
సార్ అరుణ్ సార్ మ్యూట్ చేసేసుకుంటున్నారు సార్.
అయినా కాదు మనకు షేర్ చేస్తే
కాకినాడ నుంచి షేరింగ్ ఉంది, అతను ప్రాబ్లం లేదు, కాకినాడలో షేర్ చేసేస్తున్నారు.
సార్ గుడ్ ఆఫ్టర్నూన్ సార్. శ్రీనివాస్ గారు, హలో సార్. అంటే మన సైడ్ నుంచే షేర్ చేస్తే బెస్ట్ కదా.
చేస్తున్నాను సార్, మన సైడ్ నుంచి షేర్ చేస్తానంటే సార్ ఆడియో రావట్లేదు. ఒకసారి ఎన్ఎస్ లో కనెక్ట్ అవుతున్నాను సార్, చేసి పెడతాం సార్కి. ఎన్ఎస్ లో కనెక్ట్ చేసి చేస్తాం
సార్. ఎనీ డెస్క్ లో సార్ స్క్రీన్ షేర్ చేయకుండా పీపీటీ షేర్ చేసేద్దాం. సార్ నెక్స్ట్ అని చెప్తుంటే స్లైడ్స్ మార్చేద్దాం. అది సులభం కదా సార్.
అదే
స్క్రీన్
easy.
మన
అదే
అది
easy.
ఈజీ సార్. పవర్ పాయింట్ ప్రెజెంటేషన్ పెట్టించేసుకోండి సార్. లేకపోతే నాకు పెట్టించేసుకొని మీరు సార్ నెక్స్ట్ అంటున్నప్పుడల్లా మీరు స్లైడ్స్ మూవ్ చేస్తూ ఉండండి, సరిపోతుంది. ఇన్స్టెడ్ ఆఫ్ షేరింగ్ సార్ స్క్రీన్, you can share the PPT directly and move the slides. Thank you.
Sir.
Sir, Professor Arun Kumar, sir. Hello.
Sir. Okay, okay.
చెప్పండి చెప్పండి ఓకే
Sir, good afternoon. Sir, you can keep in unmute. We can present your PPT, sir.
Yeah,
You can
just
you
1
can
minute
you can straight away deliver your lecture, and you can ask us for next slide.
Okay, sir. You're projecting there?
Yeah, I will present, sir.
Okay, fine. Right. Thank you. Thank you.
Meanwhile, you can start, sir. Just I will share your screen.
Yeah, sure.
But sir, please unmute yourself, video and audio. Very often you are muting yourself. Your audio and video is also
No, the screen is there. There's
Okay,
an issue streaming. That's why I'm
maybe network issues
so
there,
that
sir. Maybe network issues there.
yeah, network.
Yeah.
Right. Can I start?
Yeah, you can start. Meanwhile, you can start. I will share your presentation.
Yeah, do I get to see the presentation?
Yeah, just a minute, sir. Just a minute.
సార్ మాకు ఇంకా రాలేదు కదా, మీ దగ్గర నుంచి ఒకసారి షేర్
ఒక్కసారి ట్రై చేస్తాను, ఉండండి. మళ్ళీ షేరింగ్ మళ్ళీ ఒక్కసారి ట్రై చేస్తాను.
చేయాలంటే మన దగ్గర నుంచి మళ్ళీ దీంట్లో కొంచెం టైం పడుతుంది. మీ దగ్గర నుంచి ఏం చేశారు సార్ ఆల్రెడీ అన్మ్యూట్.
Sir, I think it's your network issue, but still, if you have mobile data, you can use that also.
I'm already using mobile data.
Okay. Yeah. Now it is coming, sir. Now it is coming.
ఇప్పుడు చూడండి, వస్తుందా?
రాలేదు సార్, ఇంకా ఒకసారి. It is sharing but not visible.
సార్ మేము ట్రై చేస్తాం సార్. మేము మెయిల్ పెట్టారు సార్.
నా మెయిల్ కా?
సరే సార్.
వాట్సాప్ రావట్లేదా సార్? శ్రీనివాస్ గారు, డాక్టర్ శ్రీనివాస్ గారు, హలో. ఎందుకు మనం చేద్దాం కాదు, ఎందుకు మన మెయిల్ ఐడి చెప్తాం సార్, దాన్ని ఫార్వర్డ్ మళ్ళీ అది ఓపెన్ చేసి పెట్టేద్దాం. ఈజీగా దీంట్లో ఓపెన్ చేసుకోవచ్చు.
అరుణ్ సార్, మీరు షేర్ చేస్తే స్క్రీన్ కనబడుతుంది, కాకపోతే మీరు మీ మొత్తం డెస్క్టాప్ ఫుల్ స్క్రీన్ షేర్ చేస్తున్నారు సార్. అలా కాకుండా మీ పీపీటీ స్లైడ్ షో మోడ్లో పెట్టి మీరు స్క్రీన్ షేర్ చేసినప్పుడు మీ స్లైడ్ షోని సెలెక్ట్ చేయండి సార్.
సరే మేడం, అలా కూడా చేశాను. ఐ ట్రై వన్స్ మోర్. సో యా
సార్ ప్లేస్మెంట్స్ వెళ్ళారు కదా, ప్లేస్మెంట్స్ వెళ్ళారు.
APCC.
సార్ ప్లేస్మెంట్స్ వెళ్ళాడు ఏపీసీసీ మెయిల్ పెట్టండి సార్.
gmail.com.
అంతే సార్.
వాట్సాప్ రాదంట.
130 MB ఉంది టోటల్.
సార్ ఒకసారి మీరు లాగౌట్ అయ్యి మళ్ళీ లాగిన్ అయ్యి ట్రై చేయండి సార్.
రైట్ సార్, I'll do that.
Yeah, it is, I think, possible now.
Sir, I think it is completely internet issue, sir.
Yeah, internet issue.
స్టార్ట్ అవ్వలేదు సార్ ఇంకా ఇక్కడ కూడా scientific so download first అది ఓపెన్ అయింది ఓపెన్ అయ్యే సబ్మిషన్ ఓపెన్ అయింది డౌన్లోడ్ అయిపోయింది అంతా ట్యాగ్ అయినా సరే అదే download చేస్తాను
Now, are you able to see? Are you able to see now?
Yes, sir, but your presentation is not visible. You are visible.
So there is some technical issue and because I am able to see.
Maybe network issue, sir. Maybe. Sir, you please give access. You are not given any access to your presentation. You give access to all and share it again. Your presentation. Sir, we received your presentation, but it is not accessible to us. Please give access to all.
The email Yeah, I gave access.
Thank you, sir.
అంటే దాంట్లో చాలా ఉన్నాయి సార్ పవర్ పాయింట్
Okay.
Can't I just create a Google Meet link and share? Is that not possible?
No, not required, sir. I am downloading your presentation now.
Okay, right.
We can present from here.
Okay, fine. You had any technical issues like this, or this is the first time you're having?
It is first time, sir. Maybe the issue is at your end, sir. That is network issue. Because the presentation is it is more
Normally,
than.
I attend all meetings. We share PPTs. That is never an issue here.
Maybe, sir, your presentation is 134 MB. That's why it is not opening here.
Now you have downloaded, sir, the entire presentation.
Yeah, it is going down, sir. You can start now. Give a moment. I can share it.
Yeah. And what is the time that I can take from now on? 1 hour, 1.5 hour?
1 hour, 1.5 hours, sir.
Okay, fine, right.
Now, I request NRC Kakinada, NRC Vishaka woman, and to introduce Professor Arun Kumar sir. Once again, please, because at that time, sir is not there. Oh, Rega madam.
Okay, sir.
సార్ ఒకసారి అక్కడ డౌన్లోడ్
Good
చేసి
afternoon, sir. This is Dr. Samuel David Ross from NRC Visakhapatnam. I deem it a great privilege to introduce Dr. Kota Arun Kumar Garu, all the participants of TOT on single major subjects in zoology, biotechnology and biochemistry. Dr. Kota Arun Kumar Garu has been the alumnus of University of Hyderabad since his masters and finished his doctorate at University of Hyderabad itself in plasmodium in murine models. He is postdoc from New York University School of Medicine, USA, on pre-erythrocytic stages of plasmodium using rodent models. Now he is working as assistant professor in animal biology, School of Life Sciences, University of Hyderabad. His work continues in plasmodium itself, where he is trying to prevent clinical malarial symptoms, which has implication in producing attenuated vaccines. With this backdrop, I request Arun Kumar Garu address the participants, and we are ready to hear you, sir. Thank you very much, sir. Namaste.
Yeah. Thank you for your introduction. I'm just eagerly waiting to see the presentation. If I can see, then I'll start.
డౌన్లోడ్ చాలా స్లోగా ఉంది అవుతుంది అదే మా దగ్గర డౌన్లోడ్ అవుతుంది చేస్తాం వన్స్ ఇది అయినాక Sir, is it visible, sir, now?
Yeah, it is visible.
Yeah, you can please, sir. Sorry.
No
Because
problem, sir.
the file is very large, sir. We could not able to download in proper time. You
It's
can
fine.
proceed now, sir. You can proceed
Yeah,
now.
thank you.
Welcome, sir. Welcome.
So good afternoon to all of you, and sorry for all the technical delay. So let me start the topic for next 1 hour 15 minutes. I would like to talk about two plasmodium proteins that we have identified recently, and we show that these proteins are basically required for maintaining the shape of the parasite and the infectivity of the parasite. So the topic is not going to be anything unusual for you. I will try to keep it very simple, and so that everybody can understand. So we all know about plasmodium. We all know about plasmodium, that it is a disease caused by a protozoan parasite. And can you go to the next slide, please? If you look at the life cycle, can we have the next slide?
Srinivas sir, next slide and move to please.
Yeah, I got it, sir. Yeah. So if you look at the life cycle of the parasite, you see that it is basically a digenetic parasite, meaning that it requires two hosts to complete its life cycle. The definitive host is a female Anopheles mosquito. And when female Anopheles mosquito injects humans, one form of parasite called sporozoids, they get injected into the skin. And from there, they make their way directly into what is called as the liver. And in the liver, within the hepatocytes, they undergo asexual reproduction, and they transform into what is called as the liver stages or exoerythrocytic forms. So after a brief replication in the hepatocytes, they rupture, and then they initiate what is called as the blood stage infection. And this blood stage infection basically has a number of different stages, like the ring stage, the trophozoites, the schizoids, and finally, when the schizoids rupture, they either reinitiate the blood stage infection or they transform into sexually dimorphic forms called as the male and female gametocytes. And please remember that these male and female gametocytes, they do not develop any further in the humans because of the temperature and the pH of the blood. But at this point of time, if a female Anopheles mosquito feeds on the person who is carrying these gametocytes, then there's a possibility that these gametocytes are ingested by the mosquito. And within the mosquito gut, the gametocytes, they emerge to form gametes, male and female gametes. And these male and female gametes fuse to form a zygote. The zygote transforms into a motile eukainate. It breaches the midgut epithelium, settles on the hemocoel side of the epithelium, and then undergoes a process of sporulation. And after sporulation, the oocyst rupture releases sporozoites into the hemocoel. And from the hemocoel, the parasites invade the salivary glands. So this is a very complex life cycle of malaria parasite. And most of the time when we get malaria, we are given certain drugs that is going to inhibit the parasite in the blood stages. But the main problem with such an approach is that we are developing, the parasites are developing resistance to most of the anti-malarial drugs. So in market, you have a couple of anti-malarial drugs and we know their mode of action. But the problem is that the parasites are acquiring resistance to these drugs, and therefore these drugs are no longer valid to cure malaria. So the need of the hour is to come up with vaccines that could somehow inhibit the so-called the transmission of the malaria parasites. So when you talk about vaccines, one of the best stages for developing vaccines for malaria parasites are these sporozoid stages, which I just showed you, are injected into the skin by the mosquitoes. And they'll not get into much detail, but just tell you that if you have very good antibodies against the sporozoid stages, there is a possibility to neutralize the infectivity of the parasite, which means that whatever load of parasite has been injected into the skin, more than half of it gets neutralized with the antibodies. And whatever 50% unneutralized parasites are there, they get into the liver, and then they can elicit an excellent cell-mediated immune responses. So because of the fact that parasites can, if we have antibodies against extracellular sporozoids, or cell-mediated immune response against the parasites that grow within the hepatocytes, the feasibility of developing vaccines against these stages, which we call as pre-erythrocytic stages. What do you mean by pre-erythrocytic stages? These are basically the stages they appear before the onset of blood stage infection. So therefore, our goal in our lab is to inhibit the activity of the sporozoites before they can get into the liver, or inactivate the sporozoites by some method, genetic method, so that they experience a block in the liver and therefore they are not able to initiate blood stage infection. But nonetheless, as long as they are getting attenuated in the liver, they form an excellent source of antigen for eliciting a cell-mediated immune response. So this is the approach we are taking in the lab, and towards this end, we are genetically modifying a malaria parasite such that it experiences some kind of a defect in its ability to transmit malaria. So with this premise, let me now go on and tell you a little bit more about some of the interesting features of the Plasmodium. The Plasmodium, basically, the genome was decoded in 2002, and decoding genome is very important for any organism because you get to know what are the proteins that are encoded by the genome. And once you know what kind of proteins are encoded by the genome, you know what pathways are existing, what pathways are common in humans, what pathways are common in parasite, what are unique in humans, what are unique in the parasite. And if you know that there are certain unique metabolic pathways in the parasite, then it means that these are very good drug targets because the target is unique in the parasite, the metabolism, the pathway operates in the parasite is unique and has got nothing to do in humans. There is no common mechanism of action. Therefore, whatever goes on in the parasite can be selectively targeted without affecting the host. Therefore, genome sequence is a very important landmark in Plasmodium research. Once the genome sequence was out, people started mining the genome sequence either to find the drug targets or the vaccine targets. The parasite has got 14 chromosomes, and if you see the size of the genome, it is something like 22.9 megabase, and totally the parasite encodes for nearly 5,000 genes. Now, please remember that out of these 5,000 genes, nearly 43% of the genes are called as hypothetical proteins, which means that we do not know what is the function of these 43% of the genes. Now, having known that Plasmodium is such a challenging disease, and not knowing 43% of the genes encoded by the parasite is not really a good news, because you're actually not fully exploiting the entire genome to target the parasite. Okay, which means that one has to understand what is the biology of each of these genes, which constitutes around 43% of the bulk of the genes of the parasite, which are not yet characterized. So in first half of the talk, I'm going to talk about one gene that I have selected from this pool of 43%. We have selected it because it is hypothetical, it's not annotated, it does not have any resemblance to the human gene sequences. It is totally unique to Plasmodium. And another interesting thing is that within Plasmodium, it is highly conserved, which means that if I start to make a strategy, a therapeutic approach to target that gene, probably the same could hold true when I target the other Plasmodium genes. So which means that if you're working with a conserved gene, which means you selected a gene and this gene is there in other orthologs of Plasmodium, then it is a better drug target or a vaccine target as compared to a gene that is uniquely expressed in one species of Plasmodium. Sir, can we go to the next slide? Yeah. So if you look at the life cycle of malaria parasite, nearly it took 100 years to put the different pieces of jigsaw together. So initially, the idea that mosquitoes could have done could have something to do with the transmission of malaria was proposed by Sir Patrick Manson, and Sir Ronald Ross was a student of Sir Patrick Manson. So he proposed the idea that there could be a link between malaria and mosquitoes, because they observed that mosquitoes go to the water bodies, and they come back again to the human. So they didn't know the connection that they're actually going back to the water bodies to lay eggs there, and those eggs are laid only after taking a blood meal. But nature has used that vehicle, mosquitoes, to transmit malaria was not deciphered at that point of time. Later, Sir Ronald Ross reasoned that the blood meal that mosquitoes are taking, they could contain parasites. He didn't exactly coin the term parasite, but he assumed that there could be a possibility that the mosquitoes that are taking a blood meal could carry along with the blood the agents required for malaria transmission. This is what was proposed by Sir Ronald Ross, and he has done exhaustive, experiments by feeding the chickens and humans with, mosquitoes, and then dissected the guts on day 14. And finally, he could show that there is something called as a exflagellation happening, and this process is involved in sexual reproduction of the plasmodium, and this sexual reproduction exclusively happens within the gut of the parasite. So he did most of this work in a military hospital in Hyderabad. There's one military hospital adjacent to the old Begumpet airport, and if you happen to come down on August 13th, which happens to be 12th or 13th, which happens to be the Ronald Ross day, the museum is open, and you can see all the exhibits there, which shows how he has observed this gut of the mosquitoes, how he has drawn the diagrams of exflagellation, and how he confirmed that indeed mosquitoes are agents for transmitting malaria. And then there were two other people. One is Sir Charles Laverne, and he was a person who observed the malaria parasites in the blood. And two persons, Henry Short and Percy Grenham, these are the people who said that there is a stage called as the liver stage also associated with malaria. And only after the parasite has undergone development in the liver, actually the blood stage starts, which is associated with most of the clinical symptoms. So to put all pieces of the jigsaw together to say that sporozoites enter the liver, they develop there, they initiate a blood stage infection, and then the cycle goes on in that way, it took nearly 100 years. So what we know today as a disease caused by mosquitoes, it was very difficult to even think that mosquitoes were the agents 100 years back. Next slide, please. So if you look at the gross malaria pathology, why do you get the disease? You get the disease because the parasites are within your RBCs. And while the parasites are within your RBCs, they are extensively remodeling the RBC membrane. So this remodeling of the RBC membrane is basically associated with a phenomena called as an antigenic variation. So please remember, antigenic variation is nothing but the parasite exhibiting different proteins at different stages when it is in the RBCs. And there is a family of proteins called as PFEMP1. It is called as the Plasmodium falciparum erythrocyte membrane protein 1. And this protein is a family of 59 genes. 59 genes. And you will be surprised to know that these 59 genes are not expressed at the same time. These 59 genes are not expressed at the same time, but they are mutually exclusive, which means that at any point of time, you have one gene turned on and another 58 genes turned off. Okay, so for example, if you have a wave of parasites, you can see this in the slide towards your right side corner, you can see several different waves indicated on a graph here. So if you think that the red color wave is one generation of parasite, this generation of parasite is going to have one kind of an antigenic makeup where one protein out of the 59 genes are turned on. And by the time antibodies are raised, you can see these Y-shaped structures. By the time antibodies are raised against this particular protein, you have another wave of parasite with a different antigenic makeup. And now this wave of parasites is no longer, will be neutralized with the parasite, with the antibodies that are produced against the previous strain of the parasite. So likewise, every generation of the parasite is going to produce a certain new protein, and antibodies are generated against that protein. But antibodies against the previous wave of infection are no longer valid in clearing the new wave of parasites that come up. So this phenomena is called as antigenic variation. And when these Plasmodium parasites put these antigenic variants on the surface of the parasite, on the surface of the RBCs, the entire RBC is going to become very sticky in nature, meaning that they achieve certain phenotypes to have cytoadherence with the endothelial cells which line your blood vessels. So shown here are the endothelial cells and the receptors that are present on the endothelial cells, like the VCAM, ICAM, thrombospondin, E-selectin, chondroitin sulfate A. These are all the receptors that are present on the endothelial cells, and you will see that most of the parasite proteins introduced in the RBC membrane, they are complementary in nature, interacting with these endothelial receptors, which means that when an infected RBC circulates in the blood, it has a tendency to stick with the endothelial receptors, and thereby it gets sequestered within the deep blood vessels of the RBCs. So because of this phenomena, we call that the parasite exhibits a kind of pathophysiology, and if this pathophysiology exceeds a certain limit, it is going to even sequester the micro vessels in the brain of humans, leading to a condition called as the cerebral malaria. So anything to alter the status of the infected RBCs is to do with the extensive protein trafficking happening to this membrane, and the proteins that reside here belong to the PFEMP1 family of members. Next, please. So some of the antimalarials that are present in the market are quinine, chloroquine, pyrimethamine, atavocone, and artemisinin. And the mode of action for most of these antimalarial drugs are known, but nonetheless, the parasite is acquiring resistance to most of the antimalarial drugs. So these days, these antimalarial drugs are given in a combination therapy rather than a single dose therapy. So, but nonetheless, these malarial drugs are not very effective because of the resistance the parasites are acquiring. So, I'll not go much into the details of that. Next slide, please. So, coming to the vaccines, there are several vaccines, depending upon the different stages of the life cycle of malaria parasite. And the most advanced vaccine, which has reached the phase 3 trials, is called as an RTSS vaccine. And please remember that this RTSS vaccine is actually an antibody-based vaccine, which basically targets the circumsporozoid protein, which is abundantly expressed on the surface of the sporozoids. So I'll get in a minute to the structure of the sporozoids, which are the infective forms of parasite, the mammalian host, but just go back. Yeah. Please remember that the RTSS is a vaccine against the sporozoid stages of Plasmodium, and these sporozoids are infective forms of parasite to the mammalian host. And if you look at the composition of the membrane of the sporozoids, you see that the sporozoid membrane has an extensively decorated protein called as the circumsporozoid protein. Next slide. So this spindle-shaped structure which you see here towards your left is actually sporozoid, and typically the sporozoid is a eukaryotic cell, meaning that it has got all the cell organelles of the eukaryotic cell. It has got a very distinct nucleus, endoplasmic reticulum, mitochondria, Golgi, a plasma delimited by a very good plasma membrane, apicoplast, etc. A very, very interesting thing about the Plasmodium sporozoids is that it has got a highly organized apical end. It has got a very highly organized apical end, and there are 3 unique cell organelles in this apical end. The 3 unique cell organelles are a club-shaped rhophres, which you see in pink color here, and you have this light blue micronemes, and then these brown color dense granules. So please remember that most of the proteins that have to come on the surface of the parasite, I repeat again, most of the proteins that has to come on the surface of the parasite are initially stored in these organelles, and therefore these organelles are called as the secretory organelles. So these secretory organelles called the micronemes, the rophries, and the dense granules. These are a storehouse for storing an arsenal of proteins, and these proteins are discharged from the anterior end of the parasite, and most of these proteins then decorate or attach to the sporozoid plasma membrane. And please remember that most of these proteins which are attached to the plasma membrane, they act as ligands to attach to the host cell receptors, and then they invade the host cell receptors. In addition to this unique feature of having these organelles, the rophries, micronemes, and the dense granules, another interesting thing about the sporozoids is that right under the plasma membrane, there is another complex called as a actin-myosin molecular motor complex. And this actin-myosin molecular motor complex is very essential for Plasmodium because these are all invasive stages of Plasmodium. The sporozoids invade hepatocytes, the merozoites invade RBCs, the ookinids invade the midgut epithelium. So unless they are endowed with some kind of a molecular motor right under the plasma membrane, they cannot move. And the way how the entire molecular motor connects with the extracellular parasites is simply by putting transmembrane proteins in the plasma membrane. And one side of the transmembrane protein is connected to the molecular motor towards the cytoplasmic end, and the other end of the transmembrane protein projects towards the extracellular side of the parasite, and it attaches to the substrate, which could be the cells in case of in vivo settings, or it could be some kind of a substrate molecule if you're looking at the motility of parasites on a glass slide. So you have the remarkable organization of the transmembrane proteins linking to the molecular motor on the cytoplasmic side and linking to the extracellular ligands on the other side of the cell, which is the extracellular side of the cell. So with this complex organization, both the secretion of the proteins as well as the movement of the sporozoids into the cell is achieved. And that's how the parasites, which are basically injected into your skin by the mosquito bite, are able to traverse through a number of cellular layers in the skin, reach the endothelial cells of the liver, and then they get into the hepatocytes. And that's how you see how a spindle-shaped sporozoid, once it gets into the hepatocytes, it transforms into a round structure, a bulbous structure called as the exoerythrocytic form. And what is interesting about this exoerythrocytic form is that it produces something like 62,000 hepatic merozoids. It produces 62,000 hepatic merozoids from one single hepatocyte. So if you were to look at the infection of a single sporozoid entering into the hepatocyte, it is very alarming to know that one sporozoid can produce 62,000, hepatic merozoids, and each of these hepatic merozoids, once when it ruptures the hepatocytes, can get into the blood vessel and initiate a blood stage infection. So now you can understand the potential of even one sporozoid able to cause malaria. And that's why if you can target these sporozoid stages, it would be an excellent strategy to prevent clinical malaria. So we don't want to initiate blood stage infection. So ideally, what would be better would be to identify what are the surface proteins that are there on the sporozoid and test whether, if you have very good neutralizing antibodies against these surface proteins, are you able to neutralize the infectivity of the parasite or not, and whether you are able to prevent the invasion of these sporozoids into the hepatocytes would be a very good option to check. Next slide, please. So this is actually the apical end of the parasite, as I told you, and it has got unique cell organelles, the rophries, the macronemes, and the dense granules. And I have shown this. Picture. Yeah, shown here is a picture of the anterior end of the parasite and all the different so-called cell organelles, the rophries, micronemes, and the dense granules. And we can also see here the composition of these cell organelles and the kind of proteins that reside within these cell organelles. Next slide, please. So coming back to the actual topic of the work today, how we have identified a sporozoid protein that regulates the shape. So as I told you in my previous slide, 43% of the genes in Plasmodium are not annotated. So I simply took one particular gene from this pool, and this gene ID is Plasmodium PB Anka 142290. So the goal was, what happens to the parasite if I target this particular gene? And from the gene expression data, both published data as well as some preliminary experiments that we have done, we have shown that this particular gene is highly transcribed in the sporozoid stages of the parasite. So we reasoned that if this particular gene product is important for regulating the infectivity of sporozoid, can we target this particular gene and can we prevent the clinical malaria? So that was the initial idea, for selecting this particular candidate and taking it forward. Next slide, please. So as I told you, if you have a protein or a gene that is conserved across different Plasmodium species, then it is really good because you work basically with one rodent parasite, and whatever data you get, that also implies to other Plasmodium species, provided the gene sequence is similar. Because if the gene sequence is similar, most of the functional motifs that are present within the gene, they are also similar, and therefore the function could be highly conserved across all the species. So we have took this amino acid sequence and then we have aligned with other species of the Plasmodium, and you can see that when you do that, there's a great level of similarity between the rodent parasites like the Burgii and Yolii, and between the Falciparum and Vivax, which are the human parasites, you have around 76% and 81% sequence similarity. So this grossly gave us an idea that we were working with a gene that was highly conserved across all Plasmodium species. Next. Then we did some in silico prediction with this gene, and we found that the gene encodes for a protein that is basically non-cytoplasmic, which means that the protein, once that is made by the parasite, it does not reside in the cytoplasm, and it is associated with the membrane. And within the predicted amino acid sequence, we could see several domains that are associated with the membrane. So we could see that the protein is non-cytoplasmic, and then the protein very likely associates with the sporozoid plasma membrane. And these are all the different tools that we have used that would tell us the prediction of the membrane associated domain when you give the entire amino acid sequence to the database. Can we switch the slide, please? Yeah. Now, this is a strategy for replacing the so-called locus of the gene. You have, by the double homologous recombination, replacing the target gene with that of a GFP reporter and a drug selection marker. So this is a strategy for generating a mutant. If you are talking about a eukaryotic cell and if you want to generate a mutant, then you have to target a certain gene. And this targeting is by introducing in the place of the target two genetic elements. One is a GFP marker and other is a drug selection marker. So when you take the homology arms that flank the target gene and clone them on either ends of a targeting construct, that is carrying the so-called GFP reporter and the drug selection marker, by process of double homologous recombination, you can introduce the entire cassette into the genome and you can generate a knockout. So in this way, we have replaced the hypothetical gene which we have selected in the Plasmodium genome with that of a GFP reporter and a drug selection marker. So we have put both these elements because you can select the parasites based on the GFP fluorescence as well as on the drug selection. So after successful integration, we produced a knockout, and you can see that the parasites were expressing GFP, and we did all confirmations to show that the targeting of this foreign construct in the place of the target gene was successful by integration PCRs. And the first experiment that we did with these mutant parasites was to compare the infectivity of the wild type along with the mutant parasites, and we see that the mutant parasites did not behave differently with that of the wild type parasite when we put them in equal numbers in the mouse, which clearly indicated that depleting the target gene in the blood stages did not affect the propagation of parasite.
Excuse me, sir, you are muted, sir. Please unmute your ends. Professor Arun Kumar, sir, you are muted, sir. Please unmute yourself, sir.
Yeah. Now?
Starting, sir. Continue, sir. Please, sir.
Yeah. So this is a process. Now I have shown you previously, in the previous slide, I have shown you how we have transfected the parasites with a construct, DNA construct, and how we have turned a wild type parasite into a GFP expressing parasite. And this is a procedure that we do in our lab, where we start with a wild type parasites, wild type rodent parasites. We put them into mouse. We grow them around 3 to 4% parasitemia. We harvest the parasites, put a overnight culture so that one more cycle of parasite goes in, and then we enrich the schizons by a density gradient, and then collect the schizons and then electroporate the targeting construct. So the construct what carried the GFP marker and the drug selection marker would be electroporated here into these wild type parasites. And since that construct had the homology arms, which are going to direct the construct towards the target gene, you have double homologous recombination happening in the mice once the transfection is done and injected into the mouse. And then after you get drug resistant parasites, you're going to isolate these parasites, isolate genomic DNA, and see whether you have got stable integration or not. And most of the time, the stable integrations are confirmed by just looking at the expression of GFP, because most of the parasites would express the reporter, and therefore it confirms that you're working with a correct transgenic line. So this is how we did the transfection, and we got the GFP parasites. And as I told you in the previous slide, the parasites did not differ in their ability to multiply in the blood, which meant that the depleting the gene did not affect the asexual propagation of the parasite. Next slide. So when you make a mutant, you are going to analyze the phenotype of the mutant in all stages of the Plasmodium life cycle. So we finished analysis in the blood stage. In the blood stage, the mutant did not have a defect. Then we transmitted the mutant into the mosquito. The way how you do that is put the mutants in the mouse and then allow mosquitoes to take a blood meal. And once the mosquitoes has taken a blood meal, you keep these mosquitoes for 20 days in another chamber. And then on day 14, you look at the midguards. On day 18, you look at the salivary glands. In this way, you assess whether the parasites are able to complete the development. So along with that mutant parasite, you will also run another cycle, parallel cycle, which is your wild type parasite. So the wild type parasite also, we made it as a GFP. We have put a GFP cassette in a non-essential locus. So now you have a wild type, which is GFP, as well as a mutant, which is a GFP. And these are the stages. If you look at the top left, these are the zygotes and the oocynids, and you don't find any difference in the zygotes and oocynids between the wild type parasites, which is in the top panel, and the mutant parasites in the lower panel. Likewise, if you look at the oocynids, which are these spindle-shaped parasites or the banana-shaped parasites, on the top panel again are the wild type, and in the bottom panel you have the mutants. So you do not find any difference in the mutant and the wild type. If you see on the right side, these are the dissected midguards of the mosquito, and you can see all green dots on these midguards. And these dots, each of these dots refers to the oocyst sporozoites. And if you look at the abundance of the oocyst, you don't see a difference in the numbers in mosquitoes carrying the wild type parasite and in the mosquitoes carrying the mutant parasite. So the same thing is represented here in the graph, no difference in the oocyst. And then we looked at the sporulation in the bottom panel. These are the oocysts in the top panel, the wild type oocyst, and the lower panel, the mutant oocyst, the sporulation numbers, and the structure of the sporozoid. So all these indicated that the parasite did not undergo any defect. There was no phenotype manifested by the parasite in the mosquito stages as well. So we got two important leads. The parasite, the mutants did not have any effect in the blood stage. Next, when we took these parasites to the mosquito stages, we also noted that they did not have any defect in the mosquito stages. So next stage is your salivary gland stage, where from the midgut, the sporozoites are going to go to the salivary glands. Next slide, please. So this is what we have done. We have dissected in the top panel, you see, we have dissected the glands of the mosquito, and you can see that these glands are green in color simply because the parasites are colonizing the glands. The GFP expressing sporozoites are colonizing the glands. So again, if you look at the salivary glands of the top panel and the bottom panel has the mutant residing within the salivary glands. The top panel has wild type sporozoites residing in the salivary glands. So a quick look at both these panels clearly suggests that again the mutants were not compromised to colonize the salivary gland. The mutants were not compromised to colonize salivary gland, which means that again there is no defect here. And if you look at the number of sporozoites that reach the salivary glands in two independent clones of the mutant, we found that their numbers are nearly similar to that of the wild type parasite. Next slide, please. But at this point of time, we found something very, very interesting. What was it? We analyzed the salivary glands at a higher magnification. Okay, previous slide was looking at the salivary glands at a lower magnification. But in this experiment, we have analyzed the salivary glands in a higher magnification, and you can clearly see that in case of the mutants, the sporozoites have lost their regular shape when the P-Bianca 1422-90 was knocked out. Okay, so when we knocked out this gene, the parasite lost its regular shape. So I have shown you sporozoites being in a spindle-shaped structure. In case of the mutants have lost their spindle-shaped structure, and all of them have become round in nature. So this was very unusual. The next slide, please. So we took these mutant sporozoites and we stained them with a protein antibody called as the circumsporozoid protein. I told you that the circumsporozoid protein is a major protein present on the surface of the sporozoid, and it is the vaccines against this circumsporozoid protein is called as RTSs, which has reached the phase 3 trials. This is what I've told. We have antibodies for this particular protein called as the circumsporozoid protein. So you can see that when I use these antibodies for the circumsporozoid protein, the parasites are spindle shaped in the wild type, which is expected, and you can see the kind of deformity the parasites have experienced when we have knocked out the P-Bianca 1422-90 locus. So clearly, the product encoded by P-Bianca 1422-90 is critical for regulating the parasite shape. And if you deplete this gene and fail to make this protein, then the parasites are not maintaining their regular sporozoid shape. So this was a very, very, very dramatic finding. And based on this function, that the protein encoded by P-Bianca 1422-90 maintains the shape of the parasite, we call this gene as structural integrity maintenance protein. So we call this gene as SIMP, which is an acronym for structural integrity maintenance protein. So can we go to the next slide? And concomitant with the loss of the spindle shape, all the mutant parasites lost their ability to move. So please remember that whenever parasites are moving, they release a lot of antigens from them. And the release of antigens is because most of these antigens are stored in the secretory organelles, as I told you, the rough freeze, micronemes, and the dense granules. So invariably, the motility of the parasite is associated with secretion of certain antigens. And I've shown here, in case of the wild type, a lot of circumsporozoid protein is secreted by the parasite. But you can see here, all the mutants what we have isolated from the salivary glands, they do not have the regular shape. We have already seen that. On top of that, they also lost their ability to secrete proteins and perform a gliding motility. Now you can ask me how gliding motility has got anything to do with infection. So as I told you, unless the parasites are motile, they will not move from the site from where they have been injected. Please remember that mosquitoes deliver sporozoites in skin, which is an organ that is very far away from that of liver, and they have to travel to the liver. So how do these parasites manage to move from the skin, which is a site of inoculation, to the liver where they actually develop? So this is because of the molecular motor that is present under the plasma membrane of the parasite that empowers them to move. And in case of the SIMP mutants, the parasites were not able to move. How did we know that the parasites were not able to move? We collected these sporozoites from the salivary glands and we injected different doses of sporozoites intravenously into the mouse, and we looked whether they gave rise to blood stage infection or not. So you typically get a blood stage infection on day 3.5. Please remember, you typically get an infection on day 3.5, which is nothing but the time required for the sporozoites to go to the liver, complete development in the liver, break the hepatocytes, and initiate a blood stage infection. So this period is called as a prepatency period. Prepatency is nothing but the time required for appearance of the blood stages or a blood stage infection following delivery of sporozoites. So if the sporozoites are normal, 3.5 days is a prepatency that is observed in case of mice. So you can see that with mutants, when we have injected 10,000 sporozoites or even higher doses of sporozoites, we never found any prepatency period. So in two independent experiments, either introducing 5,000 sporozoites or introducing 10,000 sporozoites, the mutants did not cause infection. And the reason why the mutants did not cause infection is simply because they are not moving from their site of infection, and this is because the parasites have become non-motile in nature. Now, what is the reason why the parasites have become non-motile in nature? This could be explained by the fact that the protein that we have depleted may act as an adapter protein linking the plasma membrane and the underlying molecular motor. So this was the assumption at this point of time, but nonetheless, to prove that, we went on to do more important experiments to see where this protein SIM actually localizes. When we depleted the gene, the parasite manifested a phenotype where we saw loss of regular shape. But where is the protein actually residing and how the phenotype is getting achieved is what was the next question. Next slide, please. So I'll not go into much details. When we complement back this gene into the knockout locus, we could restore the phenotype. And these are the complemented parasites, and the sporozoites have normal shape. So clearly, this is one way of trying to restore the function and tell that whatever phenotype you're seeing, loss of shape, was because of the loss of gene and not because of any aberrant genetic changes introduced in the genome because of gene targeting strategy. So the gene targeting strategy is so specific that we have designed the homology arms and we allowed the GFP construct and the selection marker to integrate only in that locus and not in any other locus. So that was the reason why you got a phenotype. And now again, if you target the same locus and put back a wild type gene, then you should demonstrate the restoration of the phenotype. And that's what we have shown here. We have completely restored the phenotype where a bulbous sporozoid now becomes a spindle-shaped sporozoid. So this is again very important in genetics, that when you show a phenotype by making a knockout, you should also restore the knockout and show that you revert the phenotype. Next slide, please. Then we next went on to generate a transgenic parasite where we put a tag in frame with the SIM ORF. So this approach is to localize where the SIM protein is made in the cytoplasm. So when you want to localize, you can take different approaches. One can make a recombinant protein, generate antibodies, and use these antibodies on the parasites, and then localize. But since we are more doing genetic modification of malaria parasites day in and day out, what we usually do is we usually put a tag, a reporter tag, like a 3x HA tag. HA is hemagglutinin. It's a viral protein. And if you put a 3 times hemagglutinin tag in frame with the ORF, and again you target this entire cassette into the ORF, what happens is the entire ORF gets translationally fused with the reporter tag, so that the endogenous locus would drive the expression of the target as well as the XXX HA tag. Now, what is the advantage of doing this is that you don't have to generate antibodies. You have commercial antibodies available in the market. You can get them and you can just see where the protein is expressed. Next slide, please. So after making this transgenic line, we use this antibody, anti-HA, and see where, in what all stages you get to see the expression of the protein. We found the protein in the gametocytes. We found the protein in the ookinete surface. We found the protein is made in the midgut sporozoites. And most importantly, we found that the sporozoites, the salivary gland sporozoites, are making this protein abundantly. And then we could concur that because the salivary gland sporozoites are making this protein so abundantly, if you deplete this protein, you are getting a loss of shape in this particular stage. And what was more interesting about identifying this protein is that its abundance was almost 50 to 60% as that of the circumsporozoid protein. Just now I told you that circumsporozoid protein is the most abundant protein present on the surface of the parasite. And we have identified yet another new protein, which is not of course as much as CSP, but at least I could say 60 to 70% as abundant as CSP. So which means that if you have monoclonal antibodies targeting the anti-HA region of the protein, or if you can target the C terminus of the protein where we have engineered the HA tag, there could be a possibility that you can prevent the attachment of sporozoites to the hepatocytes. Next slide, please. So with the current knowledge, whatever experiments we have done, we came up with this model. You can see here that this is a phospholipid bilayer of the parasite, and embedded within the phospholipid bilayer are several known proteins, like the circumsporozoid protein, which is attached to the phospholipid bilayer by a GPI anchor. You have a protein called as a trap that is associated with the molecular motor. And we have newly identified a protein called as a SIM, and we have put a question mark here because we think that there's one more adapter protein that is linking the SIM with the molecular motor, and either if you deplete the SIM or if the SIM is not able to establish connection with any of these adapters, probably the link is lost here, and the loss of link is what is rendering the mutant parasites non-infective. So at this point of time, we ask several important questions. When you deplete SIM, is there any cytoskeleton also getting affected? Is there a membrane detachment happening, because you see a lot of membrane falling apart on the mutant parasite? And what happens to this inner membrane complex, which is just lying underneath the plasma membrane? So we asked all these questions because we wanted to know the distortion in the parasite, the loss of shape in the parasite. Is it got anything to do with the cytoskeletal organization? Is it got anything to do with the inner membrane complex organization? And is it a phenomena only associated with membrane detachment or involving the cytoskeleton? And what is the possible orientation of the SIM protein? Okay, and what is the possible orientation of the SIM protein? We have seen that the SIM protein localizes on the sporozoid membrane, but now we wanted to ask whether the protein orients towards the cytoplasmic side or towards the so-called the extracellular side. Next slide, please. So we did some very simple experiments, and one of the reagents we have used was phyloidin FITC, and this phyloidin FITC is going to bind to the actin of the parasite. So you can see that the effactin filaments are intact both in the wild type parasite and the mutant parasites. even though the mutants exhibit a lot of distortion in case of their structure. So this clearly indicated that the mutants were not experiencing any defect in the cytoskeletal organization, but it was purely a membrane-associated phenomenon. Likewise, we have also shown here that if you look at the another structure called as the microtubule organizing center, or the inner membrane complex, the inner membrane complex lies right beneath the plasma membrane. And again, we show that the inner membrane complex is not getting affected when the mutants are manifesting this aberrant phenotype. So we rule out two possibilities, that the loss of regular shape has got nothing to do with the cytoskeleton and nothing to do with the inner membrane complex, which are two scaffolds that are present below the plasma membrane. So whatever we are seeing is purely a membrane phenomena, meaning that if you deplete SIM, you are resulting in membrane detachment. You're resulting in a phenotype. It is resulting in a phenotype where there's a membrane detachment happening, meaning that SIM acts as an anchor, trying to pull the plasma membrane and attach it to the underlying molecular motor. And if you take away that linker, if you take away that adapter, the entire plasma membrane is falling apart. Next slide. This is what we have shown in the next slide, where you have an electron. Next slide, please. Electron immuno-EM that is being, these are the sections of the sporozoid. The wild type is this cross section of the sporozoid, and on the left you have the wild type sporozoid cross section. On the right side you have all the SIMP mutants cross section. So you can see that there's a typical membrane detachment happening when the SIMP is not there. So this clearly indicates that SIMP is required for establishing a tight connection between the sporozoid plasma membrane and the so-called inner membrane complex. Next slide, please. And one more interesting thing we show in our study is that we already showed that the SIMP is membrane associated, and we have generated a line, if you remember, by fusing 3xHA C-terminus to the ORF, which means that the C-terminally engineered HA tag will be extracellular if the entire C-terminus domain is outside the parasite. So in order to prove whether the HA tag is extracellular or not, we did a very simple experiment where we did not permeabilize the sporozoids and then stained for HA antibody. And we found that without permeabilization, the HA antibody gave immunoreactivity. This clearly indicated that there is a domain of SIM that is residing on the plasma membrane of the parasite, and it is getting exposed towards the extracellular side of the parasite. And this is a very, very important finding for any new protein that has been identified, because if you have an extracellular domain that is projecting out of the cell membrane, then that domain could be a very good target for antibodies. So this is what we show here. If you do not permeabilize, you still get the immunoreactivity with the HA protein. And in case of non-permeabilization, the inner membrane complex is not getting stained. So this tells you that our staining conditions are so perfect. Non-permeabilized condition means antibodies don't get inside the cell, and that's why you don't have staining of the inner membrane complex, which is below the plasma membrane. But if you permeabilize, you get to stain both the plasma membrane proteins as well as the proteins of the inner membrane complex. So this is how the staining positive controls and negative controls work well as informing us that the SIMP protein, what we show, is associated with the membrane, has got actually a C-terminal domain that can be targeted with antibodies. Next slide, please. So we went on, I'll just finish up in another 15 to 20 minutes. So we went on to, now I told you that the C-terminus of SIMP is extracellular. So we wanted to see whether the C-terminal domain is conserved in all other species, most importantly, the Plasmodium falciparum species. And when we aligned only the C-terminus of SIMP, both for the Berghii and for falciparum, we found that there was a 76% homology, which was not bad. And then we have we have generated recombinant protein of the falciparum sim and obtained the antibodies. And we found that these antibodies are now cross-reacting with the parasites. Next slide. So the antisera that we obtained for this recombinant protein, we have used on the sporozoid surface, and you can see that the sporozoid surface under non-permeabilized conditions is now getting stained with antibodies. So if you have antibodies against the falciparum domain of the protein, cross-reacting with the rodent parasite, it clearly means that the degree of conservation of the protein is high in both cases. That's why you get to see the high level of immunoreactivity. And not only are the sporozoites expressing the parasite, even the liver stages of Plasmodium, when the sporozoites transform into exoerythrocytic forms, even these stages are expressing this SIM protein. So next slide, I'll show you an importance of the entire finding. Next slide, please. So now we have shown that there are parasites, sporozoites, which are having this protein called as a SIM, and it is basically associated with the plasma membrane, and have also shown that it has got a C-terminal domain of the SIM. Now, I want to translate these studies to falciparum simply because I cannot get sporozoid stages of falciparum. It is ethically and logistically very challenging. You need to have human blood. You need to have transmit the human blood to the mosquitoes by using some membrane feeding techniques, and then get sporozoids, and there's a lot of ethical challenges. So what we have decided is to generate a rodent parasite, which we already have in our hand, which expresses a falciparum copy of SIM. So this is the way how we are going to do it. We take the locus of the wild type parasite, which is a rodent parasite, replace it with a dual selection marker, and again in the next round the dual selection marker is replaced by a falciparum version of SIM. So please don't get into the details of what this dual selection marker is. So in a two-step manner, the endogenous locus of the rodent parasite is replaced by a falciparum locus, where the endogenous SIM is replaced by the falciparum SIM. So what would be the advantage of generating this transgenic parasite is simply that you are still working with the rodent parasite, meaning that you can transmit them to the mosquito, look at the production of sporozoids, look at the liver stage development, etc. Except that the rodent parasite is now antigenically different, meaning that it expresses a copy of Plasmodium falciparum. So this now provides us with a very good tool. If you have antibodies against the falciparum SIM, you can neutralize these transgenic parasites with these antibodies, put them back into mice, and look at their prepatency period. If there's a delay in prepatency period, or if there is a complete stoppage, a block in the prepatency period, it means that the antibodies totally neutralize the sporozoids, and then they are not able to facilitate the invasion of the parasite. So this is the current approach that we are taking. We are creating a transgenic parasite. The parasite is basically rodent in nature, but would express a falciparum copy of the SIM. We are going to produce monoclonal antibodies for falciparum SIM, use those antibodies for neutralizing the sporozoids, and then see whether you achieve the neutralization of the parasites or not. So this is a bigger implication of the work. Next slide, please. So based on the importance of the entire finding, the journal editorial has actually come up with a commentary on the work that we published. So the work was published in Molecular Microbiology, and the editorial has asked two experts to write a commentary on the work that we have published, and they said that the work has got immense implication for vaccine development, simply because we have identified a protein that resides on the sporozoid plasma membrane, has a C-terminal domain that can be targeted by antibodies. And if you deplete that protein, you have a loss in the shape of the parasite, and the loss in shape is so beautifully depicted here. It is detachment of the plasma membrane without affecting the underlying inner membrane complex. So they have, if I am going to share this paper with you in the commentary as well. So if you're interested, you can read. The commentary highlights how identifying new sporozoid surface antigens could be excellent targets for vaccine development. I'll just ask the coordinator, what is the time I'm having now in my hand?
Is it okay for another 15 minutes, sir?
Sir, how much time, sir?
Another 10 to 15 minutes, is it okay, sir?
Yeah. I'll just take another 10 minutes. That's all. So the first part of the work was identifying this new protein. The second part of the work, I'll just quickly, take you through leaps and bounds, because you have heard a lot about sporozoites. Yeah, please go ahead, sir, with the slide. Next slide, please. Can you please change the slide? Next. Yeah, so here, this is the story of another. Yeah, you can stop here. Just go back by one slide. Yeah, so in the next 10 minutes, I'm going to describe another mutant parasite. The first one was the SIMP mutant. This is another mutant my another student Narahari has generated. This mutant is called as a RON6 mutant. So RON6 is basically a protein that resides in these structures which I refer to as the roftries. Roftries, as I told you, are the stored houses for secretory proteins. And again, most of the proteins that are secreted from these roftries, they arrive on the surface of the parasite, and they make the parasite very infective to the host cells. So the approach which we have taken is, what if we deplete certain components of the roftry protein? So the way how we have done was, we have selected one particular protein called as the RON6, which we know resides within the roftries, and we have depleted that gene, meaning that we produced a knockout which is going to lack the RON6 protein. And the overall idea was, if sporozoites need these proteins to establish infection, obviously the infectivity of the sporozoites should dramatically be altered if you tweak the composition of these roftry compartment. So please remember that the roftry compartment has something like 30 proteins as its composition. The roftry compartment has got 30 proteins as its composition, and we have eliminated one of the proteins called as the RON6, and I'll show you the kind of a dramatic phenotype that the parasites manifested by just depleting one protein present in this secretory compartment, what we call as the roftries. So the mutant that we generated was called as a RON6 mutant. Please go ahead with the slides. Yeah, so this slide shows how a complex is actually formed by the parasite. So you can see all the structures: green, purple, blue, red. These are all the different families of the RONs. RONs are called as roftry neck proteins. RON stands for roftry neck proteins, and you can see the complex. So many different RONs are being secreted by the parasite. And what is unique about this parasite is that it secretes these proteins. These proteins attach the host cell membrane, and again the same proteins act as a scaffold for the entry of the parasite into the host cell. So it is quite remarkable that the parasite is making its road and also walking on that road. So it secretes all these proteins, establishes a scaffold on the host membrane, and then through that scaffold it enters into the cell, gains intracellular residence within the cell. So I'll quickly move on. Most of the approaches that we have taken to generate the knockout is same. So I just keep moving on. Sir, please go forward. We did some in silico predictions, and again, we know that the RON6 was a non-cytoplasmic protein. It is a globular protein, basically. Globular means something that would be made and secreted out, mostly for circulation. Next. All these are in silico predictions, which are going to tell that, yeah, please stop here, sir. So the previous slides showed that the protein is basically non-cytoplasmic protein. Then we looked at, we created a transgenic parasite, same way like we did for the SIM, where we have put a HA tag in frame with the RON6 ORF, and you can see that these the green color dots are the HA immunoreactivity, and you can see that the HA immunoreactivity is in the roftries, which are basically the apical end of the parasite. This is a case of the midgut sporozoites which are transforming in the oocyst. But if you take the salivary gland sporozoites, you have a dramatic relocalization of the parasite from the rostries onto the membrane. So on the sporozoite membrane, you see that the RON6 now gets accumulated. So similarly, like SIMP, you also have a very good distribution of the RON6 protein on the surface of the sporozoite, and therefore even this protein could be a very good target for antibodies. Next slide, please. So this is a strategy for generating the HA tag line. So we have already seen that. Next. These are confirmations to see that the HA tag is in frame with the RON6 ORF. Next. This is the expression of RON in the asexual stages. The top panel is the schizon stage. The bottom panel is the gametocyte stage. Next. Then we took these parasites into the mosquitoes and then to look at the mosquito stages. Next. Yeah, I think these slides are getting repeated. Yeah, next. The same experiment that we did earlier with SIMP. Yeah, yes, please go back by one slide, sir. Yeah, the same experiment as we did with the SIMP. Here also we show that if you do not permeabilize the parasite, still you get a HA immunoreactivity, which means that RON resides on the sporozoite membrane, and again the C-terminal tag here for the protein is extracellular. So we confirmed this with two different monoclonal antibodies. We are not biased in our approach. We've got two different commercial monoclonal antibodies, rabbit polyclonal, rabbit monoclonal, and both were tested under non-permeabilizing conditions and permeabilizing conditions. So if you get the same pattern for non-permeabilizing and permeabilizing condition, it means that the protein is extracellular basically and having a distinct domain that is outside the cell. Next. We also see that the expression of HA when the sporozoites are put into Hep-G2 culture, which means that not only are sporozoites expressing the RON6 protein, but also the developing parasites that come from the sporozoites, they are also expressing. So you can see that this is a very small sporozoite at 6 hours. The sporozoites is becoming EEF at 12 hours, and at 26 hours, this is again transforming into a bigger structure. And all these stages are expressing the RON6 protein. Next. We further track the development in other liver stages like 36 hours, 48 hours, and 62 hours. In all these stages, you have the expression of RON6. To show that this expression is not something that is just, I'm sorry, the red color one is the HA expression. To show that the HA expression is unique to RON6 line and not to every line, we worked with another transgenic parasite where HA was tagged to another ORF, unrelated ORF, and you can see that that particular parasite line is not expressing the HA. So this clearly indicates that we have a very specific expression of HA in all stages of Plasmodium, in the sporozoid stages, in the liver stages, and in the blood stages. Next. These are the hepatic merozoites, where I told you 62,000 sporozoids, merozoites are produced. And you can see that every dot is one parasite nuclei, and every dot is having the so-called RON6 expression. And if you look at the localization of this RON6, it's typically in the rostries. So again, clearly indicating that in multiple life cycle stages, the RON6 protein is expressed. Next. Next, please. Next, I'll skip this. So because we found that the HA tag is extracellular, we got HA tagged antibodies and we did neutralization assays. And we found that at least for one of the commercial monoclonal antibodies that we have got, we were able to successfully neutralize the infectivity of sporozoites. And this neutralization was delayed by, noted by delay in prepatency. And also interestingly, whenever there was a delay in prepatency, the survival rate of the mice was also delayed. So you might see there's a lot of information here. Let me very in a simplified manner convey to you. We took the transgenic sporozoites, RON6 HA transgenic sporozoites. we incubated the sporozoites with HA antibody for 35 minutes and we put them back into mouse. And we found that when we neutralized with the HA antibodies, the RON6-HA got neutralized. And the neutralization was manifested by delay in prepatency, meaning that the non-neutralized sporozoites gave a prepatency of 3.5 days, whereas the ones which got neutralized, they gave a delay in prepatency. This clearly indicates that if you have an extracellular domain and if it is targeted by antibodies, you affect the infectivity of sporozoites. And those sporozoites which are put into the mouse, they do not kill the mouse on par with wild-type parasite simply because you reduce the virulence of the sporozoites, reduce the infectivity of the sporozoites. Therefore, you delay the onset of that stage infection. Next, please. So we went on to, go ahead. We went on next to make a knockout of the parasite. Next, please. Next. Yeah, so we then looked at, we generated a knockout of the parasite, and then I'm not going into details the same way how I have shown for the SIMP mutant. What was interesting about this mutant was the mutants manifested a very slow growth of parasite. So the green color you see here in the bottom of the graph is the growth of the mutant parasites, whereas the red one is the growth of the wild type parasite. And this is a longevity curve because the infectivity of the mutant was very slow growing. The mice that were harboring the mutants, they died over 30 days of malaria infection, whereas all the wild type mice harboring the wild type parasites, they died on day 15. So a very, very important observation was when you made a knockout of RON6, the parasites lost their virulence. They lost their ability to rapidly multiply in the blood, and because of which they actually delayed the death of the mice. The wild type mice are killed in 15 days, whereas there's a death in case of the mice that are harboring the mutant parasite. So this is a dramatic phenotype, and if you can recollect for the SIMP, we did not find anything like this in case of the SIMP mutants. The SIMP mutants did not manifest any defect in the blood stage. The SIMP mutants did not manifest any defect in the mosquito stages. It was only in the sporozoid stage. But in comparison to that, the RON6 mutants have a dramatic decrease in the blood stage infection. They are slow growing, and also they kill the mice very slowly as compared to wild type. Next. I'll just finish in 5 minutes. Next, we complemented the parasites and we could restore the phenotype. And then we checked the mutants after blood stage infection. We next looked at the burden of the mutant, the RON6 mutant in the mosquito midgut and in the salivary gland. And cutting a long story short, we did not find any difference in the oocyst number and the salivary gland load. Next. So these are the salivary gland sporozoites. The RON6 mutant made the salivary gland sporozoites, and it had same numbers as that as the wild type parasite. So till now, we only see a defect in the blood stage infection, but not in anything in the mosquito infection. Next, please. Then we did this infection studies, just like how we did for the SIM mutants. The mutants were motile here. The RON6 mutants were motile, and they were able to cause blood stage infection. But what was the difference between the two infections caused by the wild type and the mutant RON6 was that there was a delay in the prepatency. So normally all the mice became positive in day 3 in case of the wild type infections, whereas in case of the mutants, the parasites, they were delayed by, the infection was delayed by almost 8 days. So this clearly indicated that, there was the mutants, the sporozoites are somehow less infective to the hepatocytes. Next, please. Yeah, we did some real-time PCR to look at the burden of the parasites in the liver. And again, I'm not going to get into the details. We found that both at 12 hours and at 48 hours, there was a dramatic decrease in the load of the parasite as compared to the wild type when we're looking at the RON6 mutants. Yeah, next, please. Yeah, now I'll end in, go back. I'll end the slide in 2 minutes. So one of the go back by one more slide. One of the most dramatic phenotypes of the RON6 mutant is a condition called as hyperactive splenomegaly. So hyperactive splenomegaly is something that humans also experience during malaria episodes. But what is the molecular basis? What are the histopathological changes that happen in hyperactive splenomegaly is not known. So please remember that whenever we get infected with malaria or with leishmania, we get splenomegaly. This is a normal splenomegaly. But surprisingly, when we made this mutant parasite, RON6 mutant parasite, when we looked at the spleen from the mice harboring these RON6 mutants, all the mice exhibited hyperactive splenomegaly. So I can show you in the top panel, you can see the last 3 spleens taken from the animals which were harboring the mutant parasite. This next to it is the spleens taken from the wild type parasite. And the first one, a single spleen, is that from the uninfected mice. So from uninfected mice having a weight of 101 mg to the spleens coming from the infected mice having a weight of 304 mg, this itself is a splenomegaly, a 3-fold increase in the weight. But you can see how enormously the mutants are increasing the size of the spleen. This condition is called as a hyperactive splenomegaly. So making the mutant, knowing that RON6 plays an important role in maintaining the infectivity of the blood stages is one important finding. And because of the loss of virulence, these mutants are causing a chronic infection. I repeat again, because of the loss of virulence, these parasites are causing a chronic infection. And because of this chronic infection, you have a condition called as a splenomegaly, hyperactive splenomegaly. And please remember that only during chronic infections, you have a tug of war between the parasite and the host immune system. And this is happening in case of the mutants because the mutants are slow growing. They are not getting cleared. They're not killing the host. So there's a lot of time for a ping pong game, like a ping pong ball going from this side of the court, that side of the court. So you have a lot of exchange of information having between the host and the parasite, and that's what is leading to this kind of a hyperactive splenomegaly. So this is what we have found in case of the second mutant. When we depleted the RON6, the virulence of the parasite got dramatically altered. This we have noted by slow growing phenotype in the blood stages. The mutants did not manifest any defect in the mosquito stage. The oocysts were normal. The sporozoid numbers were normal. But yet again, I think the sporozoites are compromised to invade hepatocytes because there was a lot of delay in initiating the pre-patency period. And finally, when we took the spleen from the mice that survived 30 days, we found that the mutants induced a condition called as a hyperactive splenomegaly. And we did some histopathology analysis. Just next slide, please. And that histopathology analysis said that there was a loss of B-cell lymphoid follicular pattern, over destruction of RBCs as spleen by increased number of hemocytophages, and there's an excessive spleen enlargement. And we have used 2 markers, next slide, to show that there's an overall decrease in the B-cell activation and overall increase in the T-cell activation during this hyperactive splenomegaly. So I'll stop here, and I will be extremely happy to take any questions from both the first part of the talk and the second part of the talk.
I request the participants from NRCs to interact with Professor Anupam. I request the participants across the NRCs to interact with Sir. NRC Vishaka woman, please respond.
Good afternoon, sir.
Yeah, good afternoon, madam.
NRC Visakhapatnam, sir.
Yes.
Sir, I am Dr. Y. Shanti Prabha from NRC Visakhapatnam. Really, it's a great privilege. It's really a great privilege for me to propose vote of thanks today for your session, sir. It is really an excellent research work with extraordinary presentation, sir. You have just started with a brief introduction on life cycle of parasite. And you have also given us the best targets for vaccine production are sporozoites, and the goal that we have to inhibit the activity of sporozoites. And throughout the lecture, I could feel that it was really commendable research efforts are being made on changing the structure of these sporozoites, and unique pathways of metabolism in sporozoites have been identified, as well as several other aspects like 3 unique cell organelles in sporozoites, out of which they are very much essential, or how they are efficient for infectivity of sporozoites. And your lecture on gene targeting, especially the PBAG 14, 20, 20, 90 in the present study, as well as some of the important proteins like SIMS protein and the ROD6 protein, and the mutants, from comparison of mutants and
I'm not able to
wild
hear you.
types from salivary glands. Hello, sir.
NRC please check your audio. Seems sir is unable to hear.
Mutants from salivary glands lost their structure as well as gliding ability. Excellent explanation, sir. And finally, there are some conclusions which I have noted down. Amidst of so many several ethical challenges, wonderful works have been done on the SIMS, as well as some extracellular domains of the parasite that could be another perfect target to control infectivity of sporozoites, sir. Similarly, lastly, you have given the inputs on RON6 proteins. They are produced by the parasite. The way they create the scaffolding so that the parasite could walk on it easily and infect us. And then RON6 HA sporozoite neutralization also. Really, we are extremely thankful, sir, for the wonderful presentation. Thank you for giving me this opportunity, sir.
Professor Ankumar, sir, on behalf of all the NRCs across the AP, we are fortunate enough to have groundbreaking research on Plasmodium, and it's a inauguration. You are essentially basic research. Most of the research is applied on at least after infection, they will go for therapy, something like that. But your research is basic level, that means the injury has to be stopped by various means like applying the techniques of changing the shape, something like that. So we are fortunate enough, even though we have some problems, we sincerely apologize for the technical issues from our end also, sir. Once again, thank you, sir. Thank you very much for your wonderful presentation, sir. Thank you so much. So all NRCs are requested to continue presentation by KRPs.